Good morning and welcome to today's informational hearing concerning GMO foods. For the information of those in attendance, this meeting is being videotaped by the Broadcasting Office of the House Bipartisan Management Committee. This video is also being made available to the news media and for streaming on House websites. I've given a permission as well to a filmmaker documenting Representative Fleck's experiences as a legislator to videotape the proceedings this morning. I don't think they're in attendance just at the moment, but be on your best behavior when they get here. For those in attendance not familiar with meetings like this one, we have a formal agenda to follow where speakers will make a presentation and committee members will then have an opportunity to ask questions. This meeting is subject to the Sunshine Law, and we encourage public attention in as many ways as possible, either in person or online. The comments and the spoken part of the meeting will be restricted to those who are the invited guests to testify and members of the committee. I'd also ask that you take care to silence your cell phones and electronic devices, and remind that everyone in the room must be seated so that we don't make the fire marshal unhappy. So without further ado, I should introduce myself. I'm John Marr. I'm chairman of the House Committee on Agriculture and Rural Affairs, and joined by my Democratic counterpart, Chairman Joseph Petrarca. And I'll ask Ms. Geiger to take the roll.
Chairman Marr.
Present.
Representative Bloom.
Here.
Brooks. Denlinger. Here. Emery. Fee. Here. Fleck. Hahn. Here. Hickernow. Kaufman. Keller. Millard. Here. Mao. Here. Reese. Tovak. Chairman Petrarca.
Here.
Representatives Conklin. Delicia. Here. Kabulik. Kula.
Proxy.
Maravito. Here. Newman. Painter. Rossi. Wheatley. Here.
And I'll note for the record, we are in possession of leave forms from Representative Rossi and Representative Keller. Today's hearing is, or informational meeting, I'm sorry, there's a fine point of difference, is to gather information about genetically engineered foods, genetically modified organisms in our foods. Most people have heard of GMO, but I have discovered through conversations with my colleagues and constituents that not many people really know what these terms mean. I, like many of my colleagues, am a bit old-fashioned and believe in education preceding legislation. And so I thought it would be a good idea for the committee to undertake some education about genetically engineered foods and genetically GMO attributes of our foods. And we're very fortunate today to have an esteemed panel of those with high profiles who are scientists to speak about the science involved in this subject. It is my guiding encouragement to all those who are invited that the testimony should be oriented towards education and from a scientific perspective. The questions of public policy, questions of particular legislation and so forth will be undertaken at some future time. But today is dedicated towards ensuring that the members of the House Agricultural Committee and others interested will have a sound base of scientific understanding, or at least a threshold of scientific understanding, before any further consideration proceeds. Our first guest is Dr. Troy Ott. He is a professor of reproductive physiology at Penn State University, and I would invite Dr. Ott to attend to the rostrum. And oh, you're going to testify from the tabletop. Terrific. I understand you do have a PowerPoint presentation. Is that correct? You may proceed. And Dr. Ott gets a little more time than the other testifiers just because think of this as GMO 101, and we're about to get an education. So please do for us in 15 minutes or so what you do for a first semester course for your students.
Chairman Marr, Chairman Petrarca, committee members, it's a pleasure to be here this morning to talk to you about genetically modified organisms, and specifically a little bit more about genetically modified animals. Some of my other colleagues may focus a bit more on genetically modified plants. So you're right, I've been asked to cover about 10,000 years of genetic modification in 15 minutes, so I'm going to hold on to your seats and we'll see how far we get. So I think the first question that we need to ask when we evaluate genetic engineering is why genetic engineering? That's a question I get from a lot of my colleagues that aren't in science. Why would we be engineering animals and plants? Well, on the slide here, I've got a couple of points that I think help to clarify why. Everyone knows and should be aware that the planet will add the equivalent of about 2 Chinas before the end of the century. We also know that most of the good agricultural land is already in production, and the largest portion of fresh water is currently being used in agricultural enterprises. So it's pretty clear with 2 billion arriving just before 2050 that we're going to need to produce more food with fewer resources and less impact on the environment. This is a challenge of a generation in front of us. This is not an abstract challenge. This is not an esoteric challenge. This is the challenge for our generation and the generations immediately following us.
As you catch your breath, we just gotten a text from our technical people. Could you pull the mic a bit closer or speak a bit louder so that those on television and online may hear you as well?
How's that?
Sounds better.
So let's go back in time and talk about genetic engineering in the early days. And as you look at these pictures on the screen of horses and dogs and pigs of varying phenotypes, you'll understand that about the time that humans began domesticating animals, we began a process of genetic modification. The tremendous difference in the phenotype of these horses reflects generation after generation of selective mating and then choosing the offspring that had the phenotype or the characteristics that we wanted and then breeding those. And so if you look at the dog, for example, the Great Dane and the Chihuahua, there's over 100-fold difference in body size between those two. And now that we understand the genomes of these species fairly well, and we have complete gene sequences for these, we know that we've been selecting for genes that regulate growth hormone-related genes. And so this is a process that's been going on for some time now. You can see the pig that might have been the top of the show in the 1800s there, round and relatively fat, lightly muscled. And in the last 150 years, we have a pig now that is tremendously muscled, lean, efficient in growth, and efficient in converting feed resources into lean muscle. So this is genetic engineering, no doubt. It's genetic engineering by classical breeding and selection. It's time-consuming, it's imprecise, and it takes quite a bit of time to accomplish. Well, over the last 30 or 40 years, we've begun to learn a lot about what controls traits in animals, and we now understand that traits are controlled by genes. And what you see on the screen there is the genetic code, the 4 letters of our alphabet that describe how traits are expressed in animals. And if I didn't tell you, you couldn't tell me whether that sequence was from a bacterium or whether it was from a horse or whether it was from a human. The letters of life are shared among all life forms. And so as we began to learn about this, very quickly we began to understand how we might modify these sequences. And so that top sequence there is a sequence of a gene. It's just a string of letters. And once again, I would challenge anyone to tell me whether that came from a bacterium or whether that came from a pig, because the letters are the same. In the 70s, we developed some tools that we could begin to work with DNA, and those tools are best described by the common editing tools that you see on your word processor. We learned to cut DNA, we learned to copy DNA, and we learned to paste DNA. And with that knowledge, we soon understood that we could begin to go in and modify organisms, starting with bacteria, but then working up through the mammalian species as well. And we could modify those genetic sequences to introduce traits that weren't there, to fix mutations that did arise in the genome, to develop tools for medicine, and to develop tools for agriculture. So, as soon as we learned about these technologies, we began to work with plants, and then shortly thereafter we began working with animals. And many of you will recognize this famous Pennsylvania scientist, Dr. Ralph Brinster, who created the first transgenic animal in 1982. And in this particular mouse, and the mouse is shown there with two littermates. One has a gene for growth hormone inserted into it, and the other is its littermate without the gene for growth hormone. And you can see that there was a tremendous increase in growth of this transgenic animal. The team, Ralph Brinster's group at Penn, along with many collaborators from a number of institutions, were very quickly able to demonstrate that this technology worked in essentially all animals. We could go in and we could modify gene sequences, and we could get altered phenotypes like growth, shown here. And many of you may know that Dr. Brinster received the National Medal of Science in 2010, one of the highest honors given to a scientist here in the U.S. So what are some of the major uses of genetically engineered animals? Well, I've listed a couple here. Animal models for human disease, critical use for genetically modified animals, production of transplantable organs and tissues, life-saving treatments for people with organ failure, production of human or animal drugs or other products are also major uses of GE animals. And finally, food production. Now, I've put the first three in green because those things have been going on for quite some time now. Those things have been going on for quite some time now. We've been using genetically engineered animals for the three in green, and we've been developing the ability to use genetically modified animals for food production as well. So, that was a little bit of why GE. Now, what are some of the concerns related to GE? Well, some of it's related to fear of the unknown. We could lose control. Things I hear, it's not natural, and it will negatively affect my livelihood. And what we know with every new technology, and we can start back with pasteurization of milk or with microwave ovens, as people learned more about the technology, they generally become more familiar and more acceptant of the technology. So fear of the unknown is a very natural reaction, and it's probably a very smart reaction for the human race. Our job here as scientists is to try to bring the best information to you, so as you evaluate what you're going to do in your business, that you have the best information and the greatest familiarity with this topic. Now, I'm going to deal a little bit about this "it's not natural" question, because that's one I hear a lot. And so what you see on the screen in front of you is an animal. It's actually a gastropod mollusk. And this mollusk is quite interesting because it can eat algae. And when it eats that algae, it actually breaks the cells open of the algae. It takes the organelles from the algae and incorporates it into its own cells. And so basically transferring the machinery from a plant to an animal. And after it does that, it can actually live on CO2 and light. So that's an example of gene transfer between a plant and an animal, and there are others that I could present. Now, you might say, okay, that's a mollusk and those were algae. Well, what about something that's more near and dear to our hearts? On the top, you see a stylized picture of a virus. Okay, we all know what viruses are and how they infect cells. And on the bottom, okay, that individual needs no introduction. I hope or not I hope, but I think many of you may be surprised that gene transfer between virus and animals like humans goes on all the time. In fact, approximately 8% of our genomes are viral sequences. Okay, we carry gene sequences from virus that represent, of our 3 billion bases, about 8% of those. And those gene sequences are not just there sitting dormant. I will tell you that one particular gene, a gene coding for a protein called syncytin, was actually critical for formation of that baby's placenta. And that baby wouldn't be there unless it had expressed this old viral gene for syncytia and used it to form a functioning placenta. So I hope you understand that there are no species barriers to gene transfer from bacteria to viruses, from viruses to humans, from plants to animals. It occurs all the time because the letters are the same. The code of life is shared between all living organisms. Now, I'm going to go sort of a quick run through some genetically modified animals, and some of these have been around for quite some time, and you might ask the question about, well, why aren't we seeing them on our dinner plates? And that's a question that we're going to have to, we'll deal with at the very end. So this is a genetically modified carp. Carp are highly consumed in Asia, and this is a genetically modified carp, and they're consumed in Asia, and this one has a growth hormone gene from another carp that makes it grow about 4 to 6 times faster. This genetically modified carp has been available for human consumption, but it's not in production right now in China. These are a couple of genetically modified goats that produce a malaria vaccine in their milk. This was a collaboration between Texas A&M University and Revo Biologics, and these goats can be bred as a flock, and their milk can be the females can be milked, and the malaria vaccine can be harvested from their milk. Here's another genetically modified goat that produces a protein to combat diarrhea in children. This was a goat that was prepared at University of California, Davis, by a colleague of mine, and I'll mention here, because of some of the issues related to approval of this technology and the slow process here, right now this particular investigator is working to move these goats to Brazil, where they have a more clear regulatory pathway so they can get this sort of life-saving technology to the market. This is one of my favorites, and this is a genetically modified pig called the Enviro pig. This pig actually produces an enzyme in its saliva that helps it use phosphorus in plants more efficiently. So it helps it break down the phosphorus in plants, and it reduces the phosphorus in the pig's waste by about 60%. Now, phosphorus pollution is a huge issue, particularly in the Chesapeake Bay watershed, but certainly around the world. And this is a genetic modification that makes these animals more efficient. Once again, because of an unclear regulatory pathway, these animals no longer exist on the hoof, and they only exist as frozen embryos in a liquid nitrogen tank, awaiting a more clear regulatory environment where they might move forward. Here's a genetically modified chicken that has been engineered to resist transmission of bird flu. And every fall we start to think about how bad the flu epidemic will be, what strains are there, and the big reservoir for flu is in birds. So we look to Southeast Asia for their duck markets and their poultry markets. This is a genetic modification that makes those animals more resistant to spreading flu and serving as a reservoir for human infection. Here's a goat that produces one of the first approved pharmaceuticals for use in human medicine. And so from a medicine standpoint, we have actually one product that is the product of a transgenic or a genetically modified animal. This is probably the biggest one on the horizon right now, and that's the Aqua Advantage salmon. This salmon grows 2 to 4 times faster. And this salmon was presented to the FDA in 1996 to enter the regulatory process, and it has been now 18 years, about $70 million, and it's achieved all the hurdles, FDA, EPA, environmental assessment. Everything has been signed off more than 2 years ago, and yet this company who has this great technology awaits approval of this technology. So I attended a conference in Australia here in February where we discussed this, and the general consensus is that if this particular technology, which has proven to be safe and has met all the regulatory hurdles, if it's failed to be approved, that it will put a damper on this whole industry, perhaps for a generation. So I've taken you through genetic engineering, genetic modification 1, which was breeding and selection. Some of the animals I just showed you were created using transgenesis, where we introduce genes. I'm going to talk just real briefly about the future. And I say the future, it's not really, it's going on now, and that is our new tools for modifying the genome using gene editing. And you may see these terms, and so I would like you to to recognize them. These are new gene editing tools that have come to the forefront in the last probably 5 to 7 years. TALENs, CRISPRs, and zinc finger nucleases. They have great names that you'll easily be able to remember. But basically, what these things do are they are enzymes that are naturally occurring in bacteria that can sit down on the genome and make selective modifications. Just like when you go in and edit a document on your word processor. I can go into the book War and Peace, and I can go into page 743 and change the word the to the word she. That's the level of precision that these new genetic editing tools give us. And so these technologies are going to greatly improve the precision and the ease of doing genetic modifications. So here's an animal, and I'll ask you, is this a natural animal or is this an unnatural manipulation of the genome? And those of you who are cattle producers will recognize this as a Belgian blue double-muscled breed. This is a natural mutation that occurred that creates about 40% more muscle on the cattle that express it. So it's a natural mutation. But what it told us is about how muscle is regulated in animals. And using that technology and the new tools that I just told you about, scientists at Texas A&M use talons to go in and take a breed of cattle called the Nelore breed of cattle, and they made a modification. And I think if you look at that picture, you'll see the cow on the individual on the right is much more heavily muscled than the one on the left. So this is an example of how this is being used right now. Now, there are a number of other traits that are important here. For example, we know the genes that relate to whether the animal has horns or not, and many people would like to genetically remove the horns so they don't have to remove them after the animal is born. So this is a technology that's particularly amenable to genetic modification. So some of the other traits, for example, disease resistance, reduced environmental impact, growth efficiency, are all technologies that will be amenable to these new gene editing tools. So basically, with these new technologies, we can go from a concept, for example, let's genetically remove a horn so we don't have to dehorn cattle, to a live birth within less than a year. So, is the process of genetic modification risky, and what are those risks? Next to natural hybridization and selective breeding, genetic engineering is considered the safest approach to improving crop and animal traits because of its precision. Regulatory agencies in 59 countries have affirmed the safety of GM crops with almost 2,500 approvals on 25 different crops. And the majority of those approvals have been based on the safety of the food. If you look to every major scientific organization, and I've just picked a handful out here, the American Association for the Advancement of Science, the National Academy of Science, American Medical Association, the World Health Organization, every major scientific organization that looked at these technologies have said there's nothing inherently dangerous about genetic modification, that each individual crop or animal must be evaluated based on its own merit. But the process of changing the genome is not risky in and of itself. So, as you go forward and are asked to evaluate some of the science around this, there's a lot of information on the web, and if you've been out there, you'll find it. You may find it, like me, very difficult to try to sort the good from the bad. But I'm going to give you a few pointers that I use when I look at science, because this is what you're tasked to do. You have to evaluate the science and assess the risk and assess the benefits. And I've taken 4 of my favorite ones and put them on this slide. You have the whole, I think there's 8 or 10 of them in your packet that you can look at. So I would encourage you to be very wary of sensationalized headlines. Headlines that claims that one thing causes all evil or all good. One thing that says this is the cause of every disease or this is the cure of every disease. That's a sensationalized headline. And you're pretty comfortable in knowing that there's no way that that can be the case. Now, there's a lot of details about how to evaluate scientific studies. Things like no control group or no appropriate control group, or have the scientist cherry-pick the results and ignored the vast body of research that actually disagrees with what they're trying to say. But probably number 4 is probably the most important one. If I could have you remember one of these, it would be this problem of correlation and causation. Because almost all the bad science that I see uses this, where they say one thing happens and so does another thing, and therefore the one thing causes the other thing. The correlation and causation. Correlation does not imply causation. And so I make that point here with what is a fairly ridiculous example, but it's a very common one. And because I'm a professor, we're going to give you a little bit of Latin to end up my talk, and that is post hoc ergo propter hoc, and that means, loosely translated, after this, therefore because of this. And the fallacy of this approach of correlation analysis is shown in this graph, where we've correlated the increase in autism with the increase in organic food sales in the United States. Now, none of you here would believe that those two things are a cause and effect relationship. However, you will see much science where they show a line for one thing and a line for another thing, and then they try to make you believe that the one thing causes the other thing. And what I say to my students is, when it's raining outside, everyone has their umbrella up, but you can't put your umbrella up and make it rain. So my take-home message is today: the fact that a crop or animal is produced using genetic modification tells us absolutely nothing about the risks or the benefits of that plant or animal. Each GMO must be evaluated individually for its benefits and risks, and the regulatory path must be clear and science-based. GMOs are not the only solution to feeding a growing population sustainably, but they are an essential tool to that end. Thank you.
Thank you very much. And also, not just for the content, but for shoehorning so much into a short period of time. I remember back in 1975 that the National High School debate topic dealt with the allocation of scarce world resources, because Garrett Hardin and the World Watch Institute and others predicted with a high degree of certainty that by the time the world reached 6 billion in population, we would have mass starvation. There simply would not be enough food. There wasn't enough arable soil, and so on. And you really triggered a recollection of that by pointing out that at the very beginning that world population, which is now 15% higher than anybody predicted for this moment in time 30 years ago, needs to be fed.
It does.
And that 70% of our fresh water goes towards agriculture in a time when, while we're blessed in Pennsylvania with plenty of water, most of the world does not enjoy that plenitude. So that was helpful for me. We have just a couple of minutes before we need to move on to the next panel. Are there members who would like to offer a comment or question?
Quick question.
Representative Petrarca, Mr. Chairman.
Quick question, Dr. Ott. Thank you for being here. Do you have an opinion on labeling? I mean, do folks have a right to know some of the things you discussed here or not? Is it good, bad?
Yeah, so I'm here as a scientist trying to let you guys make the hard decision. I have my own personal opinions on labeling, but I think the take-home message, the first one there, is probably the best describes my opinion on this. We put things on labels that are informative, that tell consumers important information about safeties or benefits or differences. The fact that an organism is genetically modified tells me nothing about the safety or risks or benefit of that product. That's my opinion on that.
Okay, I guess the question is, do those folks have a right to know that and maybe make that decision for themselves?
Well, I think the FDA has taken a very balanced stance in terms of what we allow. If you even go in the market now, we are overwhelmed with claims and labels, natural, free range, produced this way, produced that way. The only really label that's meaningful out there right now is the USDA organic label. We know what's behind that, and we know there's a process, and that is a very effective label for those people who want to avoid this technology. Once again, I think labels have to provide information that provides provides information to consumers. A label that says this product was made using genetic modification doesn't provide any information relative to the safety, the health, or the quality of that product.
Okay. Thank you. Representative Wheatley.
Thank you, Mr. Chairman, and I really appreciate the presentation this morning. The question I had is, you had mentioned earlier about the future need that we have to find an alternative to our food because we have more people and we have to have, less, we have to produce more with less.
Correct.
Are there other alternatives outside of this process that scientists are exploring to produce that?
Yeah, absolutely. Once again, genetic modification is an important tool for science and for agriculture, but it's not the only tool. And I think some of the other speakers will talk about some other approaches. So there are an array of important technologies that can be used to improve yields, quality, while maintaining food availability, affordability, and safety. And genetic modification is one important tool in the toolbox for that.
All right, thank you.
Representative Mowell?
Thank you, Mr. Chairman. Thank you, Doctor. I appreciate you being here. Can you give me an idea of what percentage of our food, whether it's a grain food or a living organism, is genetically modified, roughly?
I think I'll defer that question. We have a couple of speakers after me that are really experts on the crop side, and most of the food you're going to be talking about is on the crop side. So if you'll maybe just wait for some experts on the plant side. I can tell you that greater than 90% of our corn, soy, cotton are genetically modified.
What about
in the U.S.?
What about animal?
Zero.
Zero.
Zero animals have been approved. Aqua Advantage salmon is the one that sits on the cusp. That will be the first approved for consumption as a food.
Okay. Well, then, as far as grain food, then it would probably be easier to label those that are not as opposed to those that are, wouldn't you say, if 90% are?
Yeah, I'm not sure what would be the best approach, but I do support an approach where information, the information that's put on labels should be valuable, and putting a GM label doesn't tell a consumer anything about the safety, quality of that product.
So if a manufacturer wanted to, there would be no problem saying non-genetically modified grains.
Yeah, and in fact, the USDA organic label currently does that. So there's plenty of space in the marketplace, and also any company, organization can currently make that claim as well. There's no restriction on them saying that this was produced without the use of genetic modification.
Thank you, Doctor. Appreciate it. Thank you, Mr. Chairman.
Thank you again very much, Dr. Ott, and you provided a terrific segue to the next expert to speak to us, who's also a professor at Pennsylvania State University. It's Dr. John Tucker, who's Associate Professor of Entomology and Extension. Well, I did notice that you had sought recognition. Unfortunately, we are trying to keep to a very challenging schedule. So, Dr. Tucker, if I understand you're going to speak from the rostrum?
I'll speak from wherever you want.
Wherever you're comfortable.
I guess I can sit over there.
Well, have a seat, make yourself at home, and thank you very much for joining us today.
I believe a different slide set needs to be up there. Oh, thanks, Troy. Chairman Marr, Chairman Petrarca, and committee members, thank you very much for the invitation to come down to Harrisburg today. My name is John Tucker. I'm an associate professor of entomology at Penn State. I have research and extension responsibilities at Penn State, so my extension responsibilities take me out to farmers. I'm supposed to spend 50% of my time interacting with farmers and trying to solve their insect pest problems. All right. I figured I'd start with some take-home messages. First is that integrated pest management is a vital way to control pests. Specifically, since I'm an entomologist, I'll talk about insect pests. But I don't think we can have crop production in Pennsylvania without IPM. Similar to Dr. Ott's points, I think genetically modified crops, particularly I'll talk about insect-resistant corn, have clear benefits for farmers, but they have weaknesses as well. It's a technology that must be used appropriately. If it's misused, then we have some kind of larger scale repercussions that I'll touch upon. All pest management tools, no matter what they are, need to be used judiciously. Again, if they're used unwisely or without some type of foresight, we'll have a problem. And blind use of any tactic for managing a pest is going to lead to resistance or other greater problems no matter what we do. So blind use of anything is a bad thing. I think we've all learned that in other contexts. So I'm going to talk about BT crops. So while Dr. Ott talked about animals, I'm focusing my efforts today on plants. BT crops refer to a specific group of plants that have been genetically modified to be resistant to insect feeding. The first BT crop was cotton, shortly followed by corn. It makes sense that cotton was the first BT crop because we don't eat it, we just wear it. It's perhaps less risky. Then corn came along. BT refers to the scientific name of a soil bacterium. The scientific name of that soil bacterium is Bacillus thuringiensis. That's where the BT comes from. For some reason, this soil bacterium, a very common soil bacterium, it's my understanding, for some reason this beast makes an insecticidal protein that kills insects. Science has yet to elucidate exactly why a soil bacterium needs to kill a caterpillar, but it has proteins that do that. So folks figured out a while ago now that if you could take that gene responsible for that toxic protein and insert it into a plant, and then when an insect feeds upon it, that insect is killed. Now this lethality is limited to a suite of insects that are susceptible to the toxin. So you can buy BT products that target caterpillars, you can buy BT products that target beetles, larvae, you can buy BT products that target mosquito larvae or other fly larvae, but it's very specific. So this stuff is put into, this gene has been inserted into corn and cotton and a couple other plant species as well. And the mode of action is fairly simple. The caterpillar in this case ingests the toxin, the protein. When that toxin enters the caterpillar, the alkaline condition of their gut triggers the toxin and activates it. That alkaline condition is, of course, specific to insects. We have acidic guts. That's why you get heartburn. I get heartburn. To my knowledge, caterpillars don't get heartburn because they have an alkaline gut. Because we don't have an alkaline gut, it doesn't seem that these compounds are activated when we ingest them. So once that's activated, the compounds bore holes in the guts of the caterpillars, and that's what kills them. These BT seeds and BT corn in particular have a great benefit to the environment. On this figure, I have a pointer here. On this figure, on the y-axis, you have the percent of acres. That's percent of corn acres in the United States over time. And you can see that blue line is BT seed or BT corn adoption rate, more or less. You can see over time it's increased steadily. And now about 2013, we had about 70% of the grain corn in the United States is grown from BT seed. Associated with that use of BT corn is that dotted line, is that in conventional insecticide use has decreased dramatically. So now where we're using an insecticide very specifically put inside a plant, no longer do we need to spray insecticides across the landscape. And that steady drop does not make clear exactly how much insecticide has been avoided. But look at that number. Because of the use of BT crops since 1996, in corn alone, we've not used about 35 million pounds of active ingredient of insecticide on Americans' farmland. It's hard to contextualize what 35 million pounds looks like, but it's on the order of 9 space shuttles worth of insecticides that have not been sprayed, that are not getting in waterways or not being sprayed by crop dusters or high-boy sprayers. So in my mind, that's only a good thing. There are 2 main targets of BT corn. They have both been called billion-dollar pests. That is, they cause growers $1 billion worth of loss and product to control them. They are the European corn borer and the Western corn rootworm. I'll talk specifically about these pests today. European corn borer is a caterpillar that bores in stems of corn, and Western corn rootworm is a beetle larva that feeds on the roots. There are different BT traits, as they're called, or different BT genes that target each of these pest species, okay? So if you went to buy corn seed today, you could buy corn varieties that just target corn borer, a corn variety that just targets rootworm, or a corn variety that targets both. So focusing on European corn borer for a moment, prior to the introduction of Bt corn, this pest species caused northeastern corn growers about $35 million a year in both yield loss and then control costs. It bores in the stalks. You can see over in that figure there's a caterpillar going up and down that stalk. That's of course going to disrupt water and nutrient flow, and if the damage is bad enough, the corn stalk will fall over. That's of course going to make yield a little bit more challenging. With the introduction of Bt corn, each Bt cornfield becomes more or less a black hole for corn borers. The adult moths that are laying eggs on these plants can't tell the difference between Bt and non-Bt plants, and 99.99% of the caterpillars that get in those fields are expected to die. Okay? So with widespread adoption of Bt corn, the European corn borer population is steadily trending downward. Again, each field is a black hole. It's kind of hard to see. I apologize for my choice of colors here. But we've been doing research in my lab, sampling European corn borer in Pennsylvania. And so I show you some data here for Williamsport, and I could provide a number of different locations, but this one just illustrated it well. This is Williamsport over 4 years. We're comparing our data to data that was collected a decade ago. And this is the number of European corn borer tunnels in a stalk. So a tunnel is equivalent to a caterpillar because a caterpillar makes a tunnel as it goes up the stalk. And back in the early 2000s, you could find 2 or so caterpillars per stalk, or 2 or so tunnels per stalk. Now it's difficult to find a European corn borer in many non-Bt cornfields. So there is non-Bt cornfields on the landscape, and if we sample those, which we did here, it's difficult to find a European corn borer. And there's this is also happening in the Midwest. That number at the bottom here is 0.04 tunnels per stalk. This provides a clear economic benefit to growers because the pest is no longer around in great abundance. This is certainly relevant for field crop growers, folks that are growing corn, but it's also relevant for vegetable growers. European corn borer happens to also be a pest of pepper production, of tomato production, sweet corn production, and other vegetables. So there's lower populations around. Those growers do not need to struggle with this pest as much anymore because the populations are going down, and that's solely attributable to widespread adoption of Bt corn. Also, we found in our research that field crop growers in Pennsylvania can gain a benefit now by growing non-Bt corn because their input costs would be lower. Non-Bt seed costs less than Bt seed. So we're advocating for growers to consider growing non-Bt corn if they have a good sense of their local insect population, because input costs are lower, profits can be profits can be higher. The other pest species I'll consider today is the western corn rootworm. The larvae of this beetle, the beetle's on the upper right there. It looks kind of like a cucumber beetle, a little bit different. The beetle larvae feed on the roots. You can see the 2 roots there, 2 plants with the root systems. Those are pretty ugly root systems, and when you have a really poor root system like that, the corn's not taking up much from the soil, and it easily falls over. So the picture on the lower right is corn plants falling down from heavy root damage. This also causes harvest difficulties. If your corn's falling over, it's difficult to pick it up with a combine. Western corn rootworm is a pest of continuous corn production. So many folks in Pennsylvania, particularly dairies, grow corn in the same field year after year after year. And the best way to manage western corn rootworm is to rotate your crops. So grow corn, soybeans, or wheat, or something like that. Some rotation is better. But there are folks in Pennsylvania that need continuous corn, and again, they're dairymen usually. And those continuous corn growers will use a soil insecticide or Bt to control their crop. It's good to note, though, that the Bt corn varieties targeting western corn rootworm are inferior to the products targeting European corn borer. So some western corn rootworm larvae are actually expected to survive when they're feeding on the corn roots of Bt plants expressing those toxins. So about 95% are expected to die, at 5% is expected to live. So this product targeting western corn rootworm is inferior to the European corn borer products. And that has led to a problem. The problem is that we now have what's been confirmed in the Midwest as resistance of western corn rootworm larvae to Bt crops. That has occurred in the Midwest and has been confirmed by scientists at Iowa State University. But there are now 12 states where there's suspected field resistance. In all these cases, the plants are producing the proper protein, but there's still heavy root feeding. And the Bt toxin is only slowing beetle development. It's not killing them. And the beetle densities and the injury is somewhat remarkable. Sadly, this problem has come to Pennsylvania. I'll stress this is suspected resistance. It'll take a number of months to confirm whether it is resistant or not. But we found suspect fields in Mifflin County, Cumberland County, and Center County, where we have heavy root feeding on Bt plants, lodged plants, and high adult populations. So this is a problem, right? But the problem is derived from the products that don't work as well as the first generation, the European corn borer population or generation of products, and then growers that need continuous corn. But I want to stress in Pennsylvania, the problem is easily avoided if you are able to rotate. Some growers say they are not able to rotate. But one problem I see is that Bt technology has fostered a silver bullet mentality, that you can just put something in the field and that'll take care of my pest problems. And the European corn borer example provides that perfectly. That product has almost worked too well. It's worked very well. But still, I come back to the fact that IPM, or integrated pest management, is the best solution. So then I'll just go briefly into a pest management challenge that's associated with Bt corn, then I'll be done. So as I mentioned, Bt corn is fairly high priced, in some cases $350 to $400 for a bag of seed. Because of that, growers need to protect that seed. Seed companies want growers to have confidence that their seed will be protected. So seed companies are now applying to those seeds insecticides. They're applied directly to the seeds. These are typically neonicotinoid insecticides. This is a class of insecticides that's gotten a lot of press because of suspected involvement in colony collapse disorder and honeybee health. These insecticides are being put on the seed to control some sporadic early season pests. But in Pennsylvania, I went the wrong way, sorry. But in Pennsylvania and the rest of the United States, the amount of insecticides being used because of this mentality with Bt corn is going off the charts. This is the same figure I showed you before, but now I've added the red line, which is neonicotinoid seed treatment adoption. So now nearly over 90% of Bt corn now comes with these insecticidal seed treatments. And this increase in use does not correspond to an increase in pest risk. Okay, so there's no more pests out there. We're just putting more things on the seed to protect it. And these neonics have negative influence on beneficial insects. They can pollute water. And on my experience, they're making some pests worse. Believe it or not, the pest species I get most phone calls about is the lowly slug. Slugs are a big problem in no-till production, no-till grain production in Pennsylvania. But our research is showing that these neonic insecticides make slug populations worse, which is a challenge for growers, because it's difficult to find untreated seed these days. So this is not a Bt problem, but it comes along with the Bt mindset that we can have a silver bullet to control our pest species. So I'll just return to the take-home messages that I hope I've conveyed. That integrated pest management is vital to managing pests. Insect-resistant corn, GM corn, has clear benefits for farmers, but it has some weaknesses, particularly when we rely on it, particularly when the product isn't up to snuff, and we rely on it perhaps too heavily. And all these technologies must be used appropriately and judiciously, and any tactic poorly used will lead to resistance or even worse problems.
Thank you very much, Dr. Tucker.
Welcome.
Representative DeLisio, did you have a question?
Thank you. Just as a point of clarification then, so that that insect-resistant corn is considered a genetically modified?
No, ma'am. Sorry if I didn't make myself clear. So the insecticides applied to seeds are not a genetic modification. It's just this is an accompanying package that comes along with a treated seed now. So it's difficult to buy Bt seed now without an insecticide layered on top of the seed. That insecticide, when the seed is planted, gets pulled into the soil. Then it gets absorbed by the plant. So the insecticide is then systemic in the plant. Anything that bites the plant gets a dose of the insecticide. It's not a genetically modified thing, but they're nearly paired now, and it's very difficult to buy Bt seed without an insecticide on it. So in effect, they go hand in hand.
Thank you.
So clearly, it is not a genetic modification.
Thank you.
Sorry.
Thank you, Mr. Chairman.
Representative Bloom.
Thank you, Mr. Chairman, and thank you, Doctor. Years ago, many, many years ago, I got to spend a summer at Penn State interning for an entomology professor where we were simulating European corn borer damage to corn at different stages of development. So this stuff is all new to me because it was a long time ago, but very interesting. My question is there any known risk to humans from the BT-treated corn or eating animals that have eaten the BT-treated corn versus are there any known risks to humans from the application of conventional pesticides and the use of conventional pesticides?
To my knowledge, there are no verified risks of eating BT corn or eating animals that have been eating BT corn. There are concerns, however, of having food that's laden with insecticide residue. So there are very strict requirements in the food marketplace of having a threshold or below of certain insecticide residues because of toxicity concerns. So, yeah.
Thank you, Doctor. Thank you, Mr. Chair.
Thank you. With respect to insecticide residues, is that, if one were to look at a kernel of corn from a conventional seed, from a BT seed, or from a BT seed treated with a neonic insecticide, what would be the difference in terms of your scientific measures of insecticide residue?
So of course, a non-BT seed that wasn't treated and didn't receive any insecticide, of course, wouldn't have any residue. A BT seed that didn't have the insecticidal seed treatment wouldn't have any insecticidal residue besides the protein that's inside the plant, but that, to my knowledge, does not have a set threshold of concern. If there was a neonic insecticide on the seed, of course you'd find residue in the plant well into the season. The main residues that are addressed, it's my understanding, by kind of EPA standards, are typically foliar-applied insecticides that are sprayed and then land on the leaf surface. In corn, they wouldn't be on the kernel itself because of the husk, but they'd be on the outside of the husk. But if you were talking about tomatoes or cucumbers or something like that, they'd be on the fruit itself.
Very good.
I'm not sure that I answered your question.
No, that's helpful. It's no matter which approach we use, it's just a matter of which insecticide profile we wind up with.
Right, and so the large decrease in insecticide use from using BT corn has largely come from a lack of need of soil-applied insecticide and foliar-applied insecticides. These neonic seed treatments are a different beast because they are systemic in the plant.
Very good. Representative Wheatley.
Thanks, Mr. Chairman, and I'm very glad to be here for your presentation. Thank you for that. First is, and I meant to ask the first speaker this, are there any studies to your knowledge that takes a look at genetically modified, either the process through grains that we're doing or seeds or with animals, and the combination and what that could do to a population of people as it relates to any real or not real impacts?
I am not aware of any study that shows
the combination of everything that might be happening in our marketplace and what it means together.
I'm sorry, sir, the combination.
So if we're talking about genetically modified organisms, you always said, so the prior speaker said over 90% is always happening in the grain market, and what you're talking about is feeding to the plants in the market, the grain market, and then this possible expansion to animals or fish. There is a certain thing, at least I think there is, a combination of things could have an impact, right? So are there anyone, or to your knowledge, have there been any studies that kind of looks at the combination of all these things that are happening and what it might do together?
I am not aware of studies that have explored the interaction between different genetic modifications in plants and animals showing a downside or or a plus side. I'm not aware of such studies, sir.
So when we hear that there are no studies or studies have shown that there are no negative impacts to humans from these genetic modifications, it's saying from a specific set of studies that's looking at a specific modification, but not the totality of modifications.
I'm not aware of a study that kind of looked more holistically. That's right. The one exception is that I understand there are some individuals in the population that can be sensitive to Bt proteins. I understand there's some allergen concerns, but my sense is that's fairly restricted and not a widespread phenomenon. But I don't know much about that at all, sir.
Thank you. Thank you, Mr. Chairman.
Representative Mirabito.
Thank you. You alluded to the honeybee. Can you expand on that a little bit?
So honeybees are suffering from a condition called colony collapse disorder, which is kind of a widespread decline in their populations. And this condition has been described as death by 1,000 cuts. So there seem to be many factors contributing to honeybee decline. Viruses, pathogens, introduced pest species that are pests of honeybee colonies. One thing on that list, though, are neonicotinoid insecticides. It's not clear what percentage of the problem they're causing, but it seems to be quite clear that they're playing a role in causing honeybee populations to suffer. You're welcome.
Dr. Tugger, thank you again very much for educating us this morning.
Thanks for having me.
We'll now turn to your colleague from Pennsylvania State University, Dr. David Mortensen, who is professor of weed and applied plant ecology. And I should mention, if you run out of things to do at the university, my yard could use your attention. Thank you for being with us today.
Thank you for having me. At the risk of not having graphics in front of you, I'm going to actually not use the slides. I'm just going to talk. The paper that was added to your packet, entitled Navigating a Critical Juncture for Sustainable Weed Management, really captures most of what I'm going to say. So if there are numbers and percents or anything like that, you would find them in the paper. But I think it might be more effective for me just to talk than talking off of slides at this point. And I'm happy to share the presentation with Carrie, who could get that out to you guys. So thank you for having me. The subject that I'll be speaking about has to do with genetically modified crops that are made to be resistant to herbicides. So this is distinctly different than what Dr. Tugger was just speaking about, where plants are made to be resistant to insects by having something called a plant-incorporated protectant that the plant manufactures once it's been transformed. In the case of plants that are developed to be resistant to herbicides, the plant has been altered genetically through gene inserts to tolerate a herbicide that would otherwise kill the crop plant, and thereby can kill the weed plants that are growing in the crop. So this idea then that John spoke about, Dr. Tugger, and that I'm speaking about are what are referred to broadly as input traits. You alter the genome of the crop plant so that it changes the compatibility of that crop plant with the inputs that would be used to manage the pests in the crop plant. 80% of the pesticides that are used in grain crop production in Pennsylvania and across the United States are herbicides. So altering the crop genome to alter the way that herbicides are used has a big impact on the way that pests, weedy pests, are managed in those crops. We call, the term commonly that you'll read about and that I'm sure some of you are aware of, and maybe some of these terms others haven't been exposed to, is called packaging. You alter the crop genome so that it's compatible with the pest management practice that's going to be performed. And in the case of weeds, the packaging results in altering the genome so that a herbicide can be sold to control weeds in that crop. So there's an incentive then to have an altered crop genome that enables the use of a herbicide that's going to control the weeds. Genetically modified herbicide-resistant crops, in response to, I think, one of the questions definitely asked earlier, has been adopted widely in the United States. In Pennsylvania, this past summer, approximately 94% of the soybeans that were grown were genetically modified, and something like 76 or so percent of the corn. And in the South, cotton adoption is high, and much of it is driven by herbicide resistance traits. The problem with, so we wrote this paper because we were concerned about this packaging and the resulting increase in herbicide use that has gone along with it. But in addition to that, the packaging has also ushered in a very serious pest resistance problem. Actually, one in which Pennsylvania has been on the receiving end pretty severely the last 2 summers, with the explosion of Palmer amaranth pigweed plants having come in from the South and Midwest by jumps on equipment and through some cattle feed that was brought in. But right now we have from none in 1995 no weedy species resistant to glyphosate, which is the herbicide that most of the crop plants have been transformed to 28 species infesting 60 million acres of cropland across the United States. This is a huge problem. Over the last, I actually don't know how far back it goes, but over the last 5 years or so, there's been quite a push to come out with what is referred to as the next generation of genetically modified herbicide-resistant crops, where additional gene manipulation is performed so that the crop is resistant to glyphosate, but also to other herbicides like 2,4-D and dicamba. These are growth regulator herbicides, and we probably don't need to get into the details of it, but they're older herbicides. And some of the concerns that we have in my lab and a number of other people working on this around the country is that we're going to see a very dramatic rise in the use of these herbicides, and not a subtle use, but a dramatic use. In fact, I was surprised to see in the recent USDA environmental impact statement that came out about a month ago that the estimates that we had used in our paper 2.5 years ago were pretty much dead on what USDA has estimated, which is a 4- to 7-fold increase in the use of 2,4-D and dicamba. These are growth regulator herbicides that actually we have a very bad track record in their use in Pennsylvania, because they come up as the 2 compounds right now in a much less used condition, as they're not approved yet, to be the herbicides that we most frequently have problems with drifting into adjacent vegetable fields, gardeners' gardens, and other sensitive crops that are sensitive to that herbicide. The environmental profile, which was, I think was the main point of having me added to the list of speakers, it was to talk some about the impacts in the environment. So during a congressional testimony in D.C. a year and a half ago, and then several congressional briefings this past spring, I was asked, why should we be concerned? And I think often this was just to elicit more discussion about this, but why should we be concerned about a 2- to 3-fold increase in herbicide use and a 4- to 7-fold increase in these growth regulator herbicides? Well, one of the reasons that we would be concerned about this is that there's been a lot of research done that documents that the proportion of the landscape that's treated with something and the amount that something is applied, so when the land gets sprayed more frequently with more stuff, there's a very clear and predictable pattern in the appearance of that stuff, in this case, things like 2,4-D and dicamba, in our drinking water, in our surface water, appearing in our adjacent crop fields and vegetable gardens. And there's quite a literature on this. From a colony collapse perspective, there's a very clear link here as well. Herbicides generally are not very toxic directly on bees for the most part, but they're designed to be toxic on plants. And as herbicides like 2,4-D and dicamba are used more widely, we clearly will expect to see a reduction in the plants that the bees go to as alternate hosts for their pollen and nectar meals. And we've spent about 5 years researching this with a team of about 10 people out in the fields looking at this. And there are very clear declines in these experimental places where we basically simulate this out in the field and then do some modeling, where we see dramatic declines in the floristic diversity and abundance. And then most recently, with some work that John Tooker and I have done with some students, declines in the pollinator activity on some of those plant species. So that's an area of concern. So as I look forward, and what we advocate in this paper, and what I think from a policy point of view is important to think about, and I'm sure it is because I've already heard some questions about alternatives, is we must, I think, look forward to an agriculture where we see the kinds of practices that are compatible with the environment. And particularly, I worked in Nebraska, where the cropping wasn't very diverse and the fields were very large, and was fascinated when I came to Pennsylvania to see the diversity of things that we do on the landscape here on a much smaller scale, that the farming practices be compatible with that diversity, and we take advantage of that diversity in managing the pest populations. So I have, we have been sometimes chastised, but sometimes lift up, depending on who was reading our paper, by using the term the transgene, or the genetically modified trait facilitated herbicide treadmill. We ask in the paper, where does it stop, with some 40 applications for patent approval right now in front of the U.S. Patent Office for the next set of these herbicide-compatible traits in the crop genome. So I close then asking for us to look to an agriculture that relies much more heavily on practices that are truly going to be sustainable, taking us forward, and those that don't have paired increases in pesticide use on the order that we're almost certain will occur with the technology of these genetically modified traits. I will end there and be happy to answer any questions that you might have.
Thank you very much, Dr. Mortensen. From a big picture perspective, if I'm understanding you correctly, you were advocating for an integrated weed management and as to how to address herbicides and so forth, that no single approach is the right approach. You need to balance and rotate. And it sounds very similar to what Dr. Tooker was saying with respect to pest management. Mm-hmm. It's this integrated approach. Mm-hmm. Am I understanding that correctly?
Yeah, definitely. We definitely are advocating an integrated approach. That's correct.
And one follow-up. I was surprised that it sounds to me that your concern with glyphosate, do I say that correctly?
Glyphosate.
Glyphosate.
Yeah. Yep.
Glyphosate. Mm-hmm. Is not so much any residue from the glyphosate, but the resistance that it inspires and has created these, its expansive use has led to resistant species, is at 28 at last count, mm-hmm. which is forcing a turn back of the clock to older pesticides which have consequences that sound undesirable, such as infiltrating into the water table and things like that. Did I capture the essence there?
Yeah, so I guess, yes, and I think that that similar to what John was saying about the silver bullet approach, this idea that one tactic, one thing works. We know that it doesn't, and we know that an integration of things does do it, and riding on the backs of a technology, a genetically engineered crop technology that winds up basically solving the problem with a silver bullet plus one, appears to be unwise going forward. I feel very strongly about that. To say that it appears unwise would be maybe an understatement of how strongly I feel about that. Yeah.
Thank you, Dr. Mortensen. Representative Millard.
Thank you, Mr. Chairman. Thank you for your testimony today. You know, a couple of years ago, I read a book called Sustainability 2.0, and it talked about the need, in not only in acres but hectares, to meet the growing population and with quality acreage being able to produce enough food to meet the needs of society. On page 82 in your paper, on the left-hand column, about 3/4 down, I just want to make a comment here that you talk about funding for research, federal research funding, and how that has pretty much been diminished or flat for a number of years, and that we're now looking to industry to pick up the tab for research. And I think that it highlights one of our missions, one of our obligations as legislators, and many of the people on this committee serve as well on the Appropriations Committee, how important it is for ag research dollars to, continue the studies. But my question to you, based on your testimony and the two that preceded you, is what's your comments on generational studies, meaning you talked a little bit about herbicides and what the long-term effects can be and the resistance to certain strains. I'd like your comment for the record on how important it is to do generational studies, and that's how I'm going to term it.
Yeah.
Thank you.
Okay. Thanks. Yeah, I think they're critically important, and they're very hard to get funding to do, but they're critically important because snapshots in time miss some of the kinds of things that Representative Wheatley was asking about before. A tendency to simplify and look at single factors rather than a more holistic look, and then a holistic look in time. We need to do much more of that kind of work, and we need to fund that kind of work. I feel strongly about that. Yeah. Yeah.
Well, thank you very much, Dr. Mortensen. We'll now conclude our first part of our panel, which featured scientists from Penn State. I'll do a little advertisement for the value of research funding for our land-grant university and its extension services. The testimony we heard today from our Penn State experts certainly combines research labs and boots-on-the-ground extension perspectives, and that's so very important to agriculture in Pennsylvania. We'll now welcome our first of those who have traveled from out of state to visit with us. Dr. Stephanie Seneff is a senior research scientist at the MIT Computer Science and Artificial Intelligence Laboratory, and has traveled from Boston to be with us. Dr. Seneff, thank you very much.
Thank you for having me. I want to first tell you a little bit about myself. I'm a MIT born and bred. I went there as an undergraduate, got a bachelor's degree in biology. My PhD, along with a master's in engineering degrees, are in electrical engineering and computer science. Only in the last about 8 years, I've turned back into biology. Now I'm pretty much almost a full-time biologist using my computer science tools to help me do the biology.
Before we go too much further, could you pull the microphone a bit closer to you
Is that better?
and speak up?
Good.
Yes,
okay.
thank you.
And so I have published over a dozen papers in the last 3 or 4 years, in peer-reviewed journal articles in the scientific literature on the relationship between toxic and chemicals and nutritional deficiencies today and the diseases that we face in Western civilization. So that's sort of where I'm coming from. You can see from my title that I am interested in glyphosate, and I have in fact devoted most of my efforts the last 2 years studying this chemical. So this is what got me started in this 8 years ago. Even then, I noticed that autism rates were going up at an alarming rate, and I did not think that it was a genetic cause if it was going up. I wanted to find what are the environmental components that are also going up in step with this disease, and to figure out if it would make sense that those things that I would find would relate to autism. Here's what we see. That upper corner there is the most recent data coming from the CDC. 1 in 68 of our children are now diagnosed with autism. What you don't realize is that's 12-year-old children today. 12. So if you go back to the kids that are being born today, you have to extend this plot by 12 years. That means you have to go up like this, and so it's off the chart. On top of that, the boys have 4 to 5 times the risk that the girls have. So now we're going to get maybe up closer to the ceiling with the numbers that are happening right now with the children that are being born today. I think this is a terrifying trend, and I think it needs to be stopped. We need to do everything we can to figure out what's causing it and then to fix the problem. So this is a paper that showed up recently. They try to explain these increases through better diagnosis. I think that's not true. I think the increase is real. I imagine even 100% of it is real. Here they're saying, well, there's still this much, nearly half, that we really can't explain even when we talk about older parents and better diagnosis. If it's an environmental cause contributing to this increase, we certainly want to find it. That's what these researchers wrote. I certainly want to find it too, and I have found the answer. 2 years ago, I learned about Roundup, and I knew then that this was the chemical I was looking for. Glyphosate is the ingredient. It has a very simple molecule that looks like that, and it is very, very toxic to all life. So this, I was happy to have Professor Ott show the plot corresponding, linking autism to organic food. Most of you would be very surprised to hear that organic food might cause autism. It's actually a very small part of the food in our society, and therefore it could only at best explain, if it were true that organic food is toxic, it could only explain a small amount of the autism, because only a few people are eating it. However, autism does explain organic. There is a cause effect there. Kids who have autism have parents who adopt an organic diet. And it's not just autism, it's all the other things that the kids are having, the allergies, the eczema, the asthma, the ADHD, all of these things are causing parents to switch to organic food because they're looking for solutions, and those parents are finding that their kids get better when they switch to organic, and that's why those two are correlated. These two are correlated too, autism and glyphosate usage on corn and soy. This is a plot. The blue boxes are 6-year-old autism diagnosis in the school system. The red line is glyphosate usage on corn and soy crops integrated over the previous 4 years. That means from the time the child is 2 to the time the child is 6. So the correspondence is absolutely stunning. The correlation is 0.997. You don't get any better than that with a p-value. So 0.05 is significant in terms of p-values. This p-value has 6 or 7 zeros before the first significant digit, e to the minus 7. That means 6 zeros, and then you get the first extremely, extremely significant correlation. Okay, so you can say correlation doesn't necessarily mean causation. You have in your handout, you have a single sheet of other diseases that are also correlated very strongly with glyphosate usage on corn and soy. All of these things are going up dramatically, including things like dementia, obesity, all of these things. I have many more than those two that I've shown you, because I've been going through systematically the CDC data to see which diseases, and once I find the diseases, I go look at the biology to try to find a plausible explanation to link those together. So here we are, the situation today, GMO Roundup Ready crops. Others have spoken before about this issue. We have these crops. They've been wonderful because we can just spray the crop with the glyphosate and the crop won't die. It's one of the very few plants that won't die. Glyphosate is a general purpose herbicide. It kills all plants with a very short exception list of these crops that have been engineered to be Roundup Ready. That includes corn, soy, canola oil, sugar beets, tobacco, cotton, alfalfa. So only a small number of crops, but a huge amount of each one is being made, and these are going directly into the processed food industry. So what's been happening, it was mentioned, amaranth, a huge problem. All these Roundup Ready crops are producing Roundup Ready weeds. They keep using more and more Roundup to compensate for that. Now they're in a situation where they're going to have to add another chemical, and that was spoken about before, 2,4-D. They're pushing to get 2,4-D on the market. And we're going to have then corn. We already have talked about BT corn. I didn't even mention on the slide, but you've got the neonicotinoids, the insect pesticide built into the seed. So the corn is going to have the BT gene, which blows up the stomach of the bugs that eat it. It's going to have the pesticide neonicotinoid. It's going to have the 2,4-D, which is a derivative of Agent Orange that was used in warfare in Vietnam. And it's going to have the glyphosate still. This is going to be a very toxic food. Our autism situation is going to probably accelerate looking to the future. So to give you some evidence, this is the percent of GM herbicide-tolerant corn crops plotted alongside the percent of the corn that was treated with glyphosate. The point being here, you can see that from about 1998 up until 2013, we have had a steady increase in the amount of corn that has this Roundup Ready gene in it, exactly in step with the increased use of Roundup. Now, they told us when this came out, Roundup would be actually used less because of this gene. Clearly, it's not true. The more you have the gene, the more you have the Roundup, exactly corresponding on the plot. So the big question, okay, if it's on there, if you're getting it in the food, who cares if it's not toxic? And Monsanto assures us that it's not toxic. I mean, should we believe them? So I've been looking at every single paper I can find on glyphosate, and I can tell you that it is toxic. And so Monsanto has argued it's not toxic to humans. That's why it's a wonderful herbicide to use, and that's because it inhibits a pathway in the plants that we don't have. This pathway is called the shikimate pathway. And since we don't have that pathway, it can't possibly harm our cells. That's the argument. Nice and simple, but it's wrong. And one of the big reasons why it's wrong is because we have a bunch of gut bacteria in our gut, and if they're not healthy, we're not healthy. And people are definitely waking up to that fact today, and many people are doing excellent research showing the link between gut health and human health. And we have a very unhealthy gut at this point in time in this country. So also these gut bacteria then produce very essential nutrients that we can't produce because we don't have the genes to make them. And those nutrients in particular, the ones that come from the shikimate pathway, are absolutely essential to us. And the shikimate pathway produces things that will allow us to make serotonin, melatonin, dopamine, folate, I mean, lots of really important nutrients and neurotransmitters that are going to be a problem if we don't have enough of them. Thyroid hormone as well. So the other thing is that there are other ingredients in Roundup besides the glyphosate that make the glyphosate much more toxic than it would otherwise be, and that are also toxic in and of themselves. And these ingredients are not considered to be part of the active ingredients, and therefore they have not been studied by the industry to judge whether they're toxic or not. These are the adjuvants that are used to make the herbicide more toxic to the plants. And so the studies that they do on glyphosate leave them out and give you a false sense of security about the danger of the glyphosate to the animals being studied. The other thing is that the effects take time, and if you look for 3 months, you won't see anything obvious. The damage doesn't happen until after 4 months because it's a slow, insidious process that erodes your health. And so only if the study's done longer than 4 months on the animals will you see it. All the studies that have been done by the industry only go for 3 months. That's how you can avoid facing the fact that it's causing a problem. So the adjuvants, here's a paper. It showed that when you add the adjuvants, you can get up to 1,000 times greater toxicity in the active ingredient, which would be the glyphosate, than you would have without the adjuvants. And this is an especially striking quote from this paper by Ménage et al. It is commonly believed that Roundup is among the safest pesticides. Despite its reputation, Roundup was by far the most toxic among the herbicides and insecticides that we tested. This inconsistency between scientific fact and industrial claim may be attributed to huge economic interests, which have been found to falsify health risk assessments and delay health policy decisions. I think this is exactly what's going on. We've become complacent with Roundup. It's all over the place. We're comfortable with it. We think it's fine. It's not. Okay, so what does it do? I talked about the gut bacteria. It's been demonstrated to preferentially kill the beneficial gut bacteria, allows the pathogens to overgrow. You get a leaky gut, you get an inflamed gut, you get problems with the gut. It interferes with a very important class of enzymes in the liver called cytochrome P450 enzymes. They're used to detoxify other toxins, so they also become much more toxic in the presence of glyphosate. And I think that might be what's going on with the bees, that they're getting weakened by the glyphosate and then they're getting killed by the neonicotinoids. It chelates important minerals. This is absolutely a huge problem, so that the kids have deficiencies in these really important nutrients that will allow their enzymes to work. Iron, cobalt, manganese, there's a bunch of these. It interferes with the synthesis of these aromatic amino acids and methionine. These are essential amino acids that we can't produce ourselves because we don't have that pathway that glyphosate disrupts. And that, as I said, leads to deficiencies in things like melatonin and serotonin. And it disrupts sulfate synthesis and sulfate transport. Sulfate is my area of expertise, and I've written in papers. You have a handout, which is a paper by myself and Anthony Samsel, that describes all the different ways in which glyphosate is linked to celiac disease, which is one of these other diseases that's going up in step with glyphosate usage on crops. So these biological effects of glyphosate align remarkably well with the complex disease profile of autism spectrum. I don't have time here to show you the slides. I have hundreds. I have hundreds of slides. You can look at videos. I have YouTube videos of presentations or interviews with people, and also papers on my website if you want to learn more about those correlations. It's remarkable. I can explain just about everything I see in autism. And autism is a very complex disorder. I can link it all to the known toxic effects of glyphosate. So here's a plot of glyphosate and AMPA. AMPA is a breakdown product of glyphosate that's equally toxic. So you consider the two combined to look at the toxicity level. This is a very interesting paper because it shows three different kinds of soy. On the left is the organic. I don't know if I have a pointer here. Is there a pointer? It doesn't matter. On the left is the organic. In the middle is the conventional, where glyphosate was used to treat the weeds. And on the right is the GMO Roundup Ready soy. What you can immediately see is that nobody's got any glyphosate except the guys on the right. And 70% of those have very high levels. Monsanto themselves have claimed that 5.6 milligrams per kilogram would be an alarmingly high level, extreme level. This is 5.6. So 70% of the Roundup Ready soy had significantly higher than this level that even Monsanto would consider to be toxic. This has been going on, and the government has not been adequately testing how much glyphosate is actually in the food. And this is why I think we need the GMO label, because you could buy the conventional soy, and it wouldn't be organic, but it wouldn't have the glyphosate in it. You cannot afford to. You might say, well, then you should label the glyphosate, but that is going to be very costly to actually measure the amount of glyphosate in all the food that's distributed. So just to say it has the GMO label, that means it probably has the glyphosate. I will choose to buy the one that is not GMO in order to protect my family. So here's another plot. I have a ton of these. As I've said, I'm going carefully through all the data from the U.S. government. Very nice data the CDC puts out that you can look at these different diseases and see how they're progressing. Sleep disorder is no surprise because melatonin, which regulates sleep, is produced directly from that shikimate pathway that glyphosate disrupts. And this one has also a very high correlation with a very significant p-value with the glyphosate. When you look at the literature, you see that kids are sick. They have stomach problems. Certainly the autistic kids do. This one shows that over 11 million hospitalizations, very big data set, showing a 49% increase over a decade when the GMO crops are being introduced in Crohn's disease in children, which is Crohn's is a very serious disease. 71% increase in ulcerative colitis diseases in the hospital discharge data. Here's another plot. This is age-adjusted deaths due to intestinal infection. Again, perfect match with the glyphosate usage on corn and soy. And so intestinal infection, of course, is a consequence of the overgrowth of the pathogens. You're seeing huge issues with kidneys these days. Many people are on dialysis. Farmers in particular have a much higher risk of kidney failure than the general population. And here you can see the red is the glyphosate usage on corn and soy. The blue is the percentage of the corn and soy that's Roundup Ready. So that's going up faster than the glyphosate, but they're surrounding the curve of this kidney failure, which I think is directly due to this exposure to this toxic chemical. Seralini published a study a few years back. This picture went viral on the web of these rats that developed these massive mammary tumors as a consequence of being fed GMO Roundup Ready feed that was treated with glyphosate. These were at the normal levels, well below the toxic levels that are well below the residue levels that are allowed, these levels that they were exposed to over the course of their entire lifetime. He didn't see any problem after 3 months. Problems started to appear at 4 months. These animals died young, and the females had these massive tumors. And this is very interesting because I know that we're having a big issue with breast cancer these days. I have 2 personal friends who are premenopausal who've just been diagnosed with breast cancer. And I've looked at the breast cancer data. It doesn't correlate well with glyphosate usage on corn and soy. In fact, it's been going down up to 2006, and then it starts going back up quite fast. There's a corner at 2006. If you simply take out, you use a model to represent hormone replacement therapy, because people understood hormone replacement therapy was causing breast cancer, and they've been gradually cutting back on that. You account for that and take that out. Now you can get a 0.94 correlation coefficient, highly significant, between breast cancer and glyphosate usage on corn and soy crops. So this is Seralini's study. Rats fed GMO corn and soy throughout the lifespan. Females got these massive mammary tumors. And by the way, breast cancer cells proliferate in the presence of glyphosate in levels of parts per trillion. A study has shown that. The males developed liver and kidney disease, which is not surprising because glyphosate clearly attacks the liver and the kidneys, and the animals died prematurely compared to the organic-fed animals. So is glyphosate in our food? I think it's all over the food, especially the processed food. Here's just a picture of a typical hamburger meal. You've got the potatoes are going to be desiccated with herbicides. The GMO canola oil probably is what's going to be used to fry them. The cheese and the meat contain derived from GMO Roundup Ready fed cows, and it gets into the muscle. That's been shown in studies. It gets into the muscle when the animal eats it. The wheat is desiccated with Roundup right before the harvest. That's probably why we have celiac disease as an epidemic today. And high fructose corn syrup, GMO Roundup Ready corn. So pretty much all over the food. I think that future generations are going to look back on this time with alarm and disbelief. And they're going to recognize, they're going to acknowledge that we were willing to destroy our children's lives for the sake of cheap food and huge profits by the agrochemical industry.
Thank you very much, Dr.
There's
Seneff.
my further explanation.
I'll remind our guests that we all appreciate your enthusiasm, your time being here. The object of the informational meeting is to present information and have some questions and answers, and it's not a football match where we're cheering. Dr. Seneff, I very much appreciate your traveling from Massachusetts to be with us. It strikes me that most of your testimony was about your concern about glyphosate. Do you have any thoughts on GMO foods other than those that are glyphosate resistant?
Yeah, no, I think that it's very difficult to tell whether the GMO itself is toxic or not. It's very hard science. I've read some of the papers. Mei-Wan Ho has some excellent papers that discuss possibilities. One can imagine really terrible things that might come from GMOs, but we haven't been able to prove that those things actually happen.
Okay.
So I don't know. But I think the specific GMO
I
in
just want to make
general
sure I had the big
for
picture.
herbicides.
Your focus is really on glyphosates.
Well, not just glyphosate, because 2,4-D is coming now. We're going to
And
get
2,4-D
2,4-D ready. So whenever you design the plant to be resistant to the herbicide, you go down this path where the weeds become resistant also, and you use more and more, and then eventually you poison the people. That's the path we're going to take
So the
with 2,4-D.
consequence of having GMO seeds that are resistant to glyphosate or
It's the resistance
2,4-D,
genes.
That's the actual use of the insecticides then.
It's
that's-
herbicides. Herbicides.
The herbicides, I'm sorry. The herbicides, which is the
Issue.
concern.
And that's, many of the GMOs.
Not the seed, but the herbicides treated to cause that seed to
The seed is able to withstand the herbicide,
Right.
and therefore it soaks it up and it gets into the food chain. That's the big issue. And that's going to be true for all the herbicides. We now have 2,4-D resistance. So 2,4-D is believed to be a much more toxic chemical than glyphosate. So we're going to have some other interesting diseases coming along once we all get poisoned with 2,4-D.
And I very much impressed that you've made the transition from electrical engineering and computer science to this area of interest. By background, I'm an accountant. I know nothing about agriculture before I became chairman of agriculture, which is a good example of the way government works. But I am very interested in the visual display of quantitative data, and I do appreciate the variety of charts that you provided, and they're interesting. But I would observe that the accountant in me, the auditor in me, would say, I see a lot of correlation, but I'm not seeing much testimony about causation. I do notice, though, however, and maybe you just passed over it quickly, on page 5 of the slides that you gave us, when you talk about, you quote a paper of yours where you say the main toxic effects of glyphosate, and you've got 6 bullet points talking about toxic effects.
Right. In fact,
is this something that in terms of causation, that you've established in the laboratory, or
no, not in the laboratory, but I mean, it's
correlation?
research. It's researching the existing literature and then connecting the dots, because you can see what glyphosate does to living systems, and then you can see what autistic kids have, and you just have to connect the dots. And there are many, many dots you can connect. And in fact, I looked at all the different herbicides and all the different pesticides. Glyphosate was the only one that had this perfect correlation with autism. So of course, I'm going to start there. I mean, you would expect that, for example, heart disease, you would think that there'd be correlation between cholesterol, high cholesterol, and heart disease, because you figure that's causative. High cholesterol causes heart disease. That correlation in these data is much, much weaker. It's not even significant. Whereas this one has a 0.997 correlation with a significance that has several zeros after the decimal point. It's pretty hard to produce a correlation like that by accident. You cannot say correlation does not mean causation. The first thing you would do when you see something like that is to look for causation.
There's certain voting
Look
patterns
for causation
that are otherwise.
from the standpoint of the actual known physiology and the known effects of the chemical. When you see that kind of connection, that only makes sense to do that. Any reasonable person would think that.
Well, and I certainly understand that high correlation. And
Well, read the papers, because I didn't print out the other one because it's very long, but the papers have 300 or 400 references from the research literature.
Okay.
You can read the paper I gave you and also the other one, which is the original Seneff and Samsel and Seneff paper. And I have other papers as well. I've published 5 papers that talk about glyphosate, and all of them probably address its link to autism as well as to many other modern diseases. It's described in my papers on my webpage, and they're all open access, so you can get them and you don't have to pay $40 to get them behind a paywall.
Thank you very much. Other members of the committee? Yes, Representative Morabito.
Thank you, and thank you for your presentation. Can you talk a little bit about the autism and Alzheimer's? In other words, if there is this correlation, why don't we see in adults symptoms similar to autism?
We do. Dementia and Alzheimer's are both going up exactly in step with glyphosate usage on corn and soy, exactly in step with autism. I think it's the same disease. It's manifested differently in different age groups. Depression and schizophrenia are also part of that same story. Basically, these chemicals are harming the brain. And I think it makes a lot of sense to me. I mean, I can get into the details about the manganese deficiency and the NMDA receptors if you like, but I think it would probably not be appropriate here. You can read my papers. It's very clear to me. I think the dementia is going to be another crisis we're going to face in the future. We're already facing it.
Can you just briefly say something about the chemical effect on the brain? The manganese and the...
Okay, well, manganese deficiency is known to be caused by glyphosate in cows. They looked at 8 different farms in Denmark, and they had extremely low manganese across the board in all the farms. Manganese is first of all, it's essential for superoxide dismutase, which protects the mitochondria from damage. And in both autism and Alzheimer's, mitochondrial damage is a key feature of those diseases. Manganese is also needed for other things in the brain that are affected by glyphosate, in particular glutamate. Glutamate is a toxic neurotoxin, and glutamate is converted to glutamine using an enzyme that requires manganese. So when manganese is deficient, that can't happen, the glutamate becomes toxic. And I think that is actually what explains the MMR vaccine relationship to autism, because MMR contains glutamate, and the autistic kid can't metabolize it. Autistic kids have high glutamate in their blood and high glutamate in their brain, and in the brain it's linked to neurotoxicity. So that's the manganese example. I can give you the example on the bacteria if you like. I can keep going. I mean, the gut bacteria get disrupted, lactobacillus gets clobbered. Lactobacillus would protect from Chlamydia difficile. Chlamydia difficile overgrows and produces precresol. Precresol is high in the blood in both autism and in both the blood and the urine in autistic kids. And it's produced by glyphosate because glyphosate kills the lactobacillus. That's because the lactobacillus really depend on manganese.
Representative Bloom.
Thank you, Mr. Chairman. Thank you, Doctor. So based on your sense of what you've described to us, what remedy do you propose if you were legislating, if you were a Pennsylvania state legislator, what
That's an excellent
would the bill that you would introduce say?
I know. Well, I would introduce a bill that says ban glyphosate. It's been done in 2 countries already.
Okay, thank you. Thank you,
Ban
Mr. Chairman.
glyphosate.
Thank you very much, Doctor. Our next educator is Dr. Chuck Benbrook, who is the manager of Measure to Measure, a farm and food diagnostics for sustainability and health at Washington State University. And Dr. Benbrook was a late addition to our panel and traveled across the country yesterday to be with us. So thank you very much.
Thank you, Mr. Chairman. My sympathies to the committee that's new to this issue. It's a sticky wicket, and passions and feelings run very deep about it. I've been dealing with the science of genetic engineering, the safety evaluation and regulation, studies of the economic effects for farmers and U.S. Agriculture, Inc. For about 35 years now, and it's really stunning to me how rapidly in the last 3 or 4 years this issue has come to dominate much public debate and concern about agriculture. I've been a pest management and pesticide use and risk and regulation expert for my whole career, and I think I've testified once or twice before you years ago, and always on pesticides. But today, most of the people that are becoming interested in agriculture and concerned about agriculture have had their interest piqued because of concern over genetic engineering. So the GE crop technology it's the new pesticides. It's a new reason why people are paying attention to what's going on in the food and agricultural arena, and certainly the issues surrounding genetic engineering is doing more to shape global agriculture than anything at this time. Enormous changes in trade flows and technology, and the way that different countries are treating GE foods and either closing their markets to imports or embracing them. Let me start with going back to Dr. Ott's useful analogy of a word processor that can cut text from a document, can copy text, and move it around in the document and then paste. I think most people that understand the genetic engineering process would agree completely with him on the cut and copy parts of the analogy. But the pasting part just isn't as precise in the world of genetic engineering. What scientists can do is they can construct a very precise transgene that has certain genetic elements, perhaps from a bacteria, from a fish, from a petunia, and they know exactly what's in that transgene because they've used these very precise cut and copy methods to put it together. Almost, you might think of it as a DNA Lego kit. And so, but they have to get this transgene into the corn plant, or into the soybean plant, or into the cotton plant, or into the apple, or into the salmon. And there's a few techniques to do that, but none of them are nearly as precise as the paste function in a word processor. The reason is, Dr. Art also talked about, the fact that mammals and other organisms do share some DNA with viruses, but let me assure you, vertebrates and mammals, us humans, we don't like viruses. Our bodies and our cells and our DNA have multiple defenses against viruses, because if we didn't, our human species would not survive. So in order to get DNA that's outside the corn genome into it, it takes some really powerful medicines, some powerful techniques. And all of the GE corn up to now has been developed in one of two ways. There's what's called a gene gun, where literally a device like a gun, the scientists coat the transgene on a small piece of metal and they shoot it into a petri dish that has the growing corn tissues. And they hope that some of the DNA that's on the little silver tungsten bits of metal are literally by force pushed into the cells of the corn plant and express themselves. And it does happen, but it's not precise. There's several problems with that method of moving foreign DNA, not from the corn genome, into the corn genome. There can often be more than one copy of the gene that's expressed. There's no control over where in the genome that transgene is expressed, and sometimes it will lodge in a part of the genome where it has some effect on other genetically controlled physiological functions. So that it's kind of the transgene itself is very precise, but the expression of the transgene and where it goes in the plant is imprecise. There's another method that's used to move foreign DNA into plants, and it has its own problems. The technology's getting better, and it will continue to get better and more precise, and that's very good, because I think it will gradually lower the risk profile. You'll hear a lot about whether there's a consensus over the safety of GE foods or not. I've been in this area for my whole career. I've read most of the published science on GE crops, and I will say and agree with Val Giddings and other folks that generally speak on behalf of the benefits and safety of GE foods, that there is a broad consensus that there's no convincing evidence now in the literature, from laboratory work and epidemiological work, that today's GE foods have triggered any serious or significant change in health. And if you read, Dr. Ott put a slide up with National Academy of Sciences, AAAS, regulatory authorities around the world. If you actually read the reports that are behind that slide, you'll see a statement roughly that says that. I've quoted one in my written comments. This is from the 2004 National Academy of Sciences report, Safety of GE Foods, Approaches for Assessing Unintended Health Effects. So this is what they say, and they say basically the same thing in other reports. To date, no adverse health effects attributed to genetic engineering have been documented in the human population. I would agree with that statement. Most scientists that study and follow this issue would agree with it. But that statement does not mean that science has proven that GE crops are safe. I know perhaps that's a subtle difference, but it's a very important one. I think one of the reasons there's so much concern about genetically engineered plants around the world, and there is indeed deep concern, is that the testing methods and science that one would want to see to critically and dispassionately assess the safety of GE foods, it simply hasn't been done. It certainly hasn't been done in the U.S., and I can explain to you why, but an increasing amount of science is being done elsewhere around the world, in China, in Japan, in Europe, in the Soviet Union, in Brazil. And not all of this science is producing results that are reassuring about the safety of this technology, particularly when you look at what is required to use it. The neonicotinoid seed treatments are an integral part of Bt corn. Without it, the trait wouldn't be economically viable. And of course, essentially all GE crops have the glyphosate tolerance or Roundup Ready gene in it and a herbicide tolerance. So it's all to evaluate the effects of GE crops on either the environment or human health. You have to look at the totality of the traits, and there is not a global consensus that genetic engineering technology poses no unique risks. There is, in fact, I think, a growing body of evidence that there are some possibly unique risks associated with this technology, and we need to get going to try to determine whether they're real or not, how significant they are. I've been advocating getting the federal government involved in the kind of responsible food safety research that we should have been doing from the beginning for 15 years, but we still, as a country, have not chosen to make that investment. I think once that work is done, I certainly hope, and I would expect that most of the genetically engineered crops out there will get a relatively clean bill of slate. I don't think they'll get a totally clean bill of slate, but like any major technology, there'll be some downsides to it and some reasons for concern. The reason that the U.S. is in such a bind over this technology is that we've chosen not to engage on the scientific issues related to the safety of GE foods. You know, back in the days of when Dan Quayle was vice president in the first Bush administration, it was deemed to be the policy of the United States that the genetic engineering of plants is the sort of an agricultural wave of the future. The U.S. was way ahead in the game scientifically and from a technical point of view, and the U.S. government said this is good stuff, good science. It's going to strengthen our agriculture. We're going to strengthen our global position as a leader in science, and so a very accommodating regulatory environment was cobbled together from existing laws, and basically GE foods have had mostly a pass from the FDA. The FDA has never deemed a GE food safe. They've never done it. You can't find a single document relative to a GE food where the FDA says, we analyzed this data, we've done our assessment, and we have concluded it's safe. They don't do that. That's not what the FDA does. They do a voluntary consultation that accepts the claims of the technology developer relative to what's called substantial equivalence. And you know, scientifically, it's really, really shallow, and it's not accepted around the world, and it's why we have a big problem. You know, with sort of a crisis of confidence in this technology, and also growing problems with trade. Glyphosate's really a hugely important topic. I'm going to do now pesticide regulation 101 in 3 minutes. Think of this as a risk cup. When the EPA evaluates a pesticide, it looks at a broad array of toxicological studies, mostly in mice and rats, to determine what kind of adverse effect in experimental animals does a a herbicide like glyphosate or an insecticide like chlorpyrifos or a neonicotinoid like imidacloprid, where's that adverse effect in the studies? And the EPA picks the adverse effect that occurs at the lowest dose level. That's called the lowest adverse effect level. And then they pick the next dose down, where there's no effect. And that's called the no observable adverse effect level. And then they apply a 100-fold safety factor to it, and that creates a risk cup. The risk cup is basically, it's an estimate of how much glyphosate Representative Millard can ingest in a day per kilogram of his body weight. Now, glyphosate has the second largest risk cup of any commercially significant pesticide on the market today. There's one herbicide that has a slightly larger risk cup. And the reason it has such a large risk cup is despite a very complex and complete set of traditional toxicological studies. Glyphosate clearly is not in the category of high-risk herbicides, and it's certainly not in the category of high-risk insecticides. And in fact, as I said, it's got almost it's got a risk cup that table salt wouldn't be much bigger. So from a toxicological point of view, based on traditional toxicology, there hasn't been much concern about glyphosate, but there's growing concern now because of some of the evidence that it can act as an endocrine disruptor, and at relatively low levels of exposure that might be relevant even to what we're getting in the diet today, it can trigger some changes in fetal development and in the development of a child's nervous system. I don't think the evidence on glyphosate in terms of developmental neurotoxicity, it's not nearly as strong as the organophosphate class of insecticides, which have a risk cup about this big. So when glyphosate came on the market in 1972, it could only be sprayed before a crop comes up or after harvest because it kills all growing green things. And most herbicides that are applied early in the crop season, they don't result in residues in the harvested crop because they've broken down and they're long gone. So the actual exposure to glyphosate, despite the fact it had this huge risk cup, in the 70s, in the 1980s, it's way down here. Huge margin of safety. Regulators around the world said, there's no reason to worry about glyphosate. Well, then comes 1996, the commercial introduction of the first Roundup-ready genetically engineered crop, soybeans, corn, and cotton. The use of glyphosate in the United States and around the world took off, especially in areas planting significant percentages of their cropland to genetically engineered herbicide-tolerant corn, soybeans, and cotton. And as a result, glyphosate went from probably maybe number 15 in terms of global herbicide use across all crops to number 1. Glyphosate is, today in 2014, it's the most there's more glyphosate sprayed today around the world than any pesticide in history. It is an incredibly ubiquitous chemical. And as a result, we are all exposed, butterflies, frogs, bacteria are exposed to glyphosate in a number of new ways over a much longer period of time. And this big risk cup, it's moving up. It might be here now, it might be here, but one of the things that's also going on is some regulatory authorities think that this cup is too big, and that there actually may be a need to cut it down by about half. And I think that despite the fact that on most traditional measures of toxicity, glyphosate is one of the least toxic major pesticides ever discovered and used, we may begin to be having a problem with the biological effects of glyphosate exposures. It is a scary thought for most Americans to realize that this is a chemical. Probably most people in this room have it in their blood. Most babies that are born, you can detect it in the blood in the umbilical cord. And the levels in our diet, in the water, in rain, in fog, in the soil, on largely a global basis, are incrementally going higher because of this proliferation of use. So I sure hope glyphosate is as safe as regulators have felt it is, but it's certainly my judgment that we really need to pick up our pace in this country in doing independent science on some of these risks, because the science is being done around the world, and it's having a huge impact on markets. And then I want to just spend a couple minutes talking about why I think your committee is not going to be able to avoid this issue. We have American agriculture is having an increasing problem with our major exports, particularly exports that involve genetically engineered crops. We've had over $1 billion in corn grain and distiller dry grain exports turned around by China in about the last year and a half over a BT corn trait introduced by Syngenta that has not been approved in China. And the Chinese have gotten very good at testing food for unimproved GE traits. And so they've discovered several boatloads of distiller dry grain and grain that has a low level of contamination with this particular BT corn gene. And the economic impact on corn growers is enormous. There's already been 4 major lawsuits filed against Syngenta, and they're looking at another hundreds of millions of dollar judgment, just as in the case of Starlink and the rice. The Bayer rice gene that created a similar disruption in our rice exports. Well, I live in the Pacific Northwest, and we have wonderful dry and hot growing conditions, perfect for alfalfa. We have water from the Columbia River. One of the big growth sectors in agriculture in the state of Washington and Oregon is high-value alfalfa. It's going to China, Japan, Korea, and other Pacific Rim markets. There's tankers coming over with cars, and they're going back with high-value alfalfa hay, and it's fetching a very good price. Well, the Chinese, having had this success in detecting this unimproved corn trait, decided they best start looking for the Roundup Ready gene in alfalfa, because we in the U.S. approved Roundup Ready alfalfa 2 years ago, and it's now started to be grown on a quite significant share of Western alfalfa, probably 60 to 70%. And this gene hasn't been approved in China, and lo and behold, they're turning around boatloads of alfalfa hay. And it's having a huge impact on this growth market. One of the next ones to come along could be apples. There's 3 really important new genetically engineered foods that you'll hear a lot about and really deserve discussion. You've already heard about the salmon, the GE salmon. There's a genetically engineered sweet corn that's been on the U.S. market. 2014 was the second year. It expresses 3 BTs and also is herbicide tolerant or Roundup Ready. And this is the first food that human beings eat in largely a raw form that contains BT proteins, GE BT proteins, and I'm very concerned about it. And it's the kind of food that definitely should be labeled if parents and pediatricians are going to have any chance of determining whether this new genetically engineered sweet corn might be contributing to corn allergies. The third high-profile new genetically engineered crop is the Arctic apple. And this is an apple that there's been really there was not an attempt to move new DNA into the apple, but rather using what's called antisense technology, the apples were genetically engineered to turn off a gene that's already in the apple genome. And these genetically engineered apples don't brown as fast. That's their primary trait. Well, the apple industry in the Pacific Northwest is not supportive of the approval and commercialization of this technology because 60% of apples from Washington State are exported, and a significant share of those exports go to countries with strict GE labeling laws, if not basically a de facto market ban on genetically engineered food. Well, it turns out that the Arctic apple, the transgene used to shut off this particular gene in the apple, the transgene included a kanamycin antibiotic marker gene. I mean, every genetically engineered crop, that transgene includes within its construct an antibiotic marker gene. And that's so the scientists, after they've moved the gene in, have an easy way to test whether that gene's being expressed. And so they use an antibiotic marker gene, and they can treat the genetically modified corn or apple or soybean plant with the antibiotic. And if the antibiotic works, then they know that the transgene has not been properly expressed. So the use of antibiotic marker genes in genetic engineering technology has always been of some concern to scientists, and the Europeans imposed a regulation that's now in effect, which basically says genetically engineered food that's going to be approved for sale in Europe can't have antibiotic marker genes. Well, this apple does, and the Chinese have taken a similar position. So there's considerable concern now in the Pacific Northwest that if this apple were approved and planted, there almost certainly will be gene flow from the genetically engineered apple trees to other apple trees, and the Chinese and others will develop a test, and they'll be able to detect it, and the Pacific Northwest will start to lose what is right now very favorable access to these high-value Pacific Rim markets because of concern over that gene. So this is the world that we live in, and regardless of the positions of the U.S. government, most developed and high-value markets around the world are concerned about GE crops. And
with all due respect, Doctor, I do need to ask you to try to wrap up.
I'm done.
Our time limit on this room is a brick wall, and we have others that need to
I got to
comments.
right to where I wanted to right at the end, and I'm glad to try to respond to any questions.
Thank you. Well, I suppose there's never been a better moment to drop the fun fact as a guy from Pittsburgh area to a fellow from Washington State with your famous apples. And we have some pretty famous apples here in Pennsylvania, but Johnny Appleseed began his trek across America from Pittsburgh, just so you know. Having gotten that on the record, I did want to go back and on two big picture things to make sure I'm getting the thrust. Overall, it sounds like you and Dr. Seneff would agree that the issue isn't actual known adverse effects of the GMO. Your concern is really about the insecticides that are welcome because of the engineering of the seeds.
No, not entirely. I am concerned about sweet corn expressing 3 different Bt proteins. The levels of these Bt proteins in this new sweet corn are fairly high. A child that might have 2 ears of sweet corn at a summer picnic is going to get a dose of these Bacillus thuringiensis proteins that has, no human being has been exposed to it at all. The lax view of Bt proteins has been based on the notion that they break down very quickly once they get into the acidic conditions of the mammalian stomach, and there's indisputable data and science over many decades that show that indeed they do break down fairly quickly. But some of the recent studies show that there are, in fact, intact Bt proteins that are getting into the human blood supply. And so it's becoming important to start to ask what other effects that might they be having. There's another thing that just recently came to light. There's a number of drugs that are administered by placing a capsule under your tongue. And the drugs dissolve, and they go directly into the bloodstream through your, the very, there's a lot of blood flow in your mouth and under the tongue, and the drugs go directly into your blood supply. And so there's now evidence that some of the Bt proteins in that sweet corn actually go directly into the blood supply through the mouth without going through the GI tract and getting broken down. And these Bt proteins, they're crystalline-like molecules, and the way that they control insects is they lodge on the inside of the stomach and cause abrasion and eventually a hole in the stomach. And that's why there's a significant concern about the lower GI tract problems that might be associated. So, it's an area that I think, new and better independent research is desperately needed.
Now, and you provided us in your paper with a excerpt that you read from the National Academy of Science study that said, to date, no adverse health effects attributed to genetic engineering have been documented in the human population. Now, that was as of 2004. Are you, as a National Academy of Science, done, 10 years is a long time in science. Has there been anything from the National Academy of Science to contradict that in the last 10 years?
The Academy came out with another report on genetically engineered foods in 2010, but it focused solely on the environmental impacts. There was no substantive discussion of health effects. That 2004 report is the last substantive assessment by the Academy on food safety issues. And I might point out, the 2004 report is before the new corn rootworm trait was introduced into corn. It was before any corn expressed more than one Bt gene. Smart Stack corn expresses 6 different Bt proteins. And there's never been any independent science done on the possible synergistic or cumulative effects of these 6 different Bt proteins in corn. And the National Academy of Sciences has never addressed substantively this issue of stacking traits, which is if you're a farmer, you know about stacked traits because the cost of that seed goes up incrementally with each trait that's added. So the stacking of traits, it raises a whole new set of scientific and regulatory issues that we simply haven't dealt with.
Thank you. Thank you very much. And we very much appreciate your traveling across the country to be with us. Our next guest has also traveled a considerable distance to be with us, Dr. Kevin Folta from the University of Florida. He's a professor and chairman of the Horticultural Sciences Department. Dr. Folta, thank you very much. You're going to speak from the rostrum?
Yeah, thank you. I think this works a little better. Is that on?
There's a button at the stem there.
There you go. Okay, there we go. Very good. Thank you very much. Thank you very much, Representative Mara, for having me along, and thank you to the committee. This is a topic I really commend the wisdom of bringing aboard scientists to take part in this discussion, because too often it's being shared among people who are not scientists in this area, and especially that you would bring them from your land-grant institution is fantastic. I've been studying this area for about 30 years. Just briefly, my PhD is in molecular biology. I've done a lot of work in this area over the years, written seminal books on the topic in genomics and genetics. My lab was the lab to sequence the strawberry genome back in 2011. All of my funding has been from public, federal and state agencies, along with some money from some strawberry companies and strawberry industry. Mostly my job is chairman of a horticultural sciences department. So what my job is, I work with 56 faculty that are all over the state. We cover all areas of plant science and in hort crop production. We don't grow much GMO in Florida. We're all your fruits and vegetables, and there's very few fruits and vegetables that are GM. So I also do a lot of on my own research program. We study how light affects plant traits and how we can use non-chemical means to extend shelf life. We also are interested in genes that affect flavor. So I know this area really well. My outreach program is in biotech and how I can help people understand how it works. I meet all the time with anti-GM activists and anti-GM groups because we have a good time discussing how the nuts and bolts of this technology work. They all ask, well, how much do you get from Monsanto? And the answer is zero. Nothing personally, nothing for research. Our department gets nothing, and our university gets a dribble out in the panhandle to study some weed stuff. But nonetheless, you take a lot of heat when you stick your head in the noose. You go out there and say, I'm going to help you understand this, and you get a lot of heat as a scientist to communicate in this area. This is from just the last week about how all my work is funded by Monsanto, or none of it is. It's part of the problem of this dialogue, is that when there's a wall of manufactured risk and manufactured artificial associations and risk about this technology, it stops the flow of the good communication of the science. We do know that transgenic technology, this GMO stuff, is a precise extension of conventional plant breeding. That the techniques used pose no more risk than conventional breeding. And that's a synthesis from the National Academy of Sciences, the AAS, AMA, many others. This is a technology that has less risk, in my opinion, than traditional breeding. We'll talk about that in a moment. 17 years of use, there has not been one confirmed case of illness or death related to these products, and that is absolutely clear. A recent study showed no change in animal health over 30 years, 15 of which was done with a 100% GM diet. There are only 8 commercial crops with about 3 traits that are being used. What I want to first touch on is the fact that nothing you eat comes from here. Everything has been changed by humans. This map shows the centers of origins of all of our major food crops, and it reminds us that everything we're growing here is an artificial situation. The crops that we eat today have wild antecedents that are virtually useless. What Mother Nature put here is not very useful. They're crops that are difficult to eat, sometimes poisonous. It was human intervention that put all these genes, that stacked the genes into favorable backgrounds that allow us to have the wonderful food options that we have today. We stand today with the most variety of food available and the most safe and healthy food supply ever known by history. And food with the definition we received the other day was GMOs are food with DNA that has been altered in a way that does not occur naturally, which I would then posit everything we eat is genetically modified by humans. This is an important table, and I won't go through all of it, but it compares and contrasts the main ways in which we genetically improve crops. We have standard hybridization, polyploidization, where chemicals are used to increase the numbers of chromosomes. We have mutation breeding, where chemicals and radiation are used to damage DNA to create new traits. We have crossing species that may never cross naturally, but humans facilitate that cross. All of these methods change tens to hundreds of thousands of genes and alleles, and nobody cares. We don't know what genes are changed. We don't know what genes are altered. We don't know what the products are. We don't know what's happening. No idea. Nobody wants a label. You can grow them in organic culture. Nobody cares. But if you change one gene that you know exactly how it works and what it does, or you change 3 genes, you know exactly what they do, where they're inserted in the genome. And I would argue with Dr. Benbrook that we know exactly where a transgene inserts. It takes an afternoon, well, a couple afternoons, and a few $1,000s to sequence a genome. And I can tell you exactly where a transgene exists in any genome. So companies today, as they're improving crops, they sequence every potential release long before it's even progressing into their pipeline. They know where the genes are inserted. But so for some reason, we're worried about this problem here, this issue here, when we insert one gene or a small number of genes, where we don't care about genetic alterations that are done at a much more haphazard level. There are only 8 biotech crops, and really just 3 main commercial traits. But these are the only 3, only 8, and many people are curious about that because they think tomatoes and strawberries are in there, and they're not. 8 currently available crops. And how do we add a gene to a plant? And this, again, plays into this perception, the fear and the creepy factor that's put out there publicly. That's the non-scientific end of this. They'll tell you that genes are shot in with syringes into crops that aren't even GMOs. Okay, this is the public dialogue, and I really want to separate the public dialogue from the scientific dialogue. How this works in science is if we can get a gene in the one cell, we can grow a new plant from it. It's a property we've known for a long time, that if you take one cell and introduce the gene, you can produce an entire plant where every cell will contain that gene. Typically, we do this with agrobacterium. A gene gun was mentioned, but it's a little bit out of favor. Agrobacterium is used to place a single gene or cassette of genes into that cell. We can grow that cell into a new plant. You could come to my lab and we could do this in a week if you like, and we could start the process. It's incredibly simple, incredibly well known. So how do the traits work? And the reason I'll talk about these, I won't go too much into it because Dr. Tooker did a nice job with BT, but how do they work? And if you understand how they work, they're not so scary. So BT is pretty cool stuff, as mentioned before. It's a wonderful insecticide used for cabbage loopers and organic culture. Great stuff. And one point that was left out that Dr. Benbrook maybe got a little bit different interpretation is that the caterpillar eats this stuff. It goes in and is processed in the alkaline gut, but it binds a specific molecule on the surface of the insect gut like a lock and key. And this is why it's so specific to specific pests. This is why a beetle one doesn't work on caterpillars, a caterpillar one doesn't work on beetles. It binds a specific receptor and causes the inside and the outside of the gut contents to mix, and it dies of septicemia. And in BT corn, we just have the corn produce the protein rather than have to apply the protein, which is why it's cost effective. And again, the advantages are decreased broad-spectrum insecticide use. The limitations, of course, that you do see pockets of resistance that require additional treatments. And no evidence has been shown that BT is dangerous. There's no evidence whatsoever that shows that it's a threat to humans, and the evidence that shows it's in the blood is tenuous at best. It was detected at below the level of detection of the kit. So if you go through that paper carefully, you find that there was nothing truly detected. Glyphosate-resistant crops are where you can apply glyphosate, as mentioned, to kill the plants that are not glyphosate-resistant, where the ones that are remain. It's a tremendous tool for farmers because it allows them to suppress weed growth and control weeds without harming their crop. And it's a very simple process. This looks complicated, but it's not. In plants, you need amino acids to make proteins, and that's done through a process through the Shikimate pathway, A to B to C, and an enzyme called EPSPS catalyzes that reaction. So if you take out EPSPS by binding it with glyphosate, a specific target of glyphosate, now you can't make proteins and your plant is dead. Bacteria have the same pathway, but a little different version of the enzyme that doesn't bind glyphosate. So if you put the bacterial version into a plant, now the plant makes glyphosate makes protein just fine in the presence of glyphosate. Pretty cool. You're switching out one component for another that does exactly the same job and now allows the plant to live in the presence of the herbicide. So the problems, again, that it allows what is absolutely a low toxicity herbicide. Glyphosate in all evaluations, even in California, has been called low toxicity or very low toxicity, saves farmers money. Of course, the weed issue is there and has been addressed. Big issue here is that this isn't a scientific debate. We don't see it in scientific conferences. We don't talk about this among scientists about, is this good or bad? What we're curious about is why can't we deploy it? That's our big hang-up. This is a social debate, and it's fueled by misinformation. The main people driving it have a stake in profiting from it. You see people who make a living selling you bad information. Adams and Shiva even recently called for the death of scientists and journalists who promoted biotechnology. The arguments, we could go down this list, but it's what we've heard already before. Arguments from ignorance. We shouldn't do it because we don't know. Arguments because everybody else does it. They claim no independent research, which is completely wrong. About a third of research is. And it matches the industry research. There's a reliance on one-off science, just one publication, or confusing correlation and causality, which we've seen in tremendous success. claims based on limited data or even no data. Some scientists say that they have claims that they've never substantiated even after 10 years. The problem is that it stops innovation. And this is what really bothers me as a scientist, and this is what scientists do debate. We know we can biofortify food. We can supply vitamin A, enriched folate, iron, and other compounds in plants and distribute those to the needy and the people that in the developing world. We have plants that can grow in heat, cold, flood, drought, salty soil, that resist pathogens and pests. We have these, but we can't deploy them because of the tremendously high burden of testing and deregulation that we have to go through in order to achieve release. It's too expensive for a guy like me to release a product. We know for consumer products that we can have low acrylamide potatoes, which are healthier, allergy-free peanuts, allergy-free wheat, and healthier oils. And there's even disease-resistant chestnut that can restore forests to how they were, and renewable fuels and paper. This is really important, and most of these products are arrested because of the fear and misinformation. I'll touch on labeling briefly, but just to kind of reiterate the ideas here is that a right to know only works if there's a willingness to learn, and that a consumer can't make an informed choice if the information is wrong or false. We need to have science stepping in here. We need to have science making this determination, not activists. One other really cool one is that labeling laws they exempt products where it's inconvenient. So cheese doesn't have to be labeled in Vermont, even though it's fermented through a GMO product, GMO rennet. So it's kind of the exceptions that make it seem a little bit disingenuous. So I'll conclude with this. The main cause of anti-biotechnology environmental groups is exaggerate risks and ignore their benefits. I'll skip over to genetically engineered cotton and corn has been on my family farm since 1996. By increasing crop yields that resist many of the problems, it would be helpful. The real losers in not pursuing this will be developing nations. Instead of decades of discovery and research, people will suffer greatly for years to come. It's crucial that they reject the propaganda of extremist groups before it's too late. Who said it? The president of Monsanto? It was President Jimmy Carter on August 26, 1998. That this technology, as judged by a scientist, an engineer, and a humanitarian, was good then, and it's just as good now. I'd be very happy to answer any questions.
Thank you, Dr. Folta. Did I understand correctly that your thesis is that in one way or another, everything we consume, everything we eat, is as a result of human intervention that has stacked the genes, to use your phrase?
Yes, everything that we consume is a result of human intervention in plant genetics. The genetics have been improved by simply breeding and selection, blindly looking at the factors or the other compounds that may be present inside those plants, not testing, not worrying about it. A lot of the selection came because it was something that didn't make us sick. So over the years, we've removed a lot of those things from our food supply. But all plants have the possibility to make interesting toxins and problems, and it has happened through traditional breeding.
Okay, now, I'm not a scientist, and I'm not a farmer, and I'm going to pick, for example, a crop that doesn't grow in Pennsylvania at all, as far as I know, which is bananas. And I think I read somewhere along the line that for the major banana producers that they have gone to this technology where every banana is genetically identical to every other banana, that there's no variation at all. And I know you're a strawberry guy.
No, that's
so
true about
maybe
bananas.
that's out of your realm, but am I remembering something there?
Oh, no, it's all in my realm. Bananas are clonally propagated from a single foundational plant, as are strawberries, as are most citrus varieties, as are apples, which is why apples are not an issue for cross-pollination. They're all produced vegetatively. So bananas are all from one single crop, which can be part of the problem why it doesn't resist disease.
And your table, I must confess, it looked very interesting, but my eyes aren't sharp enough to make that out. Maybe you could make that available to us in
Absolutely.
our copy.
It's actually, if you Google Frankenfood paradox, I'll be happy to supply it otherwise, but for anyone else who's here, it shows in comparison contrast the degree of radical genomic alteration that occurs in our different forms of genetic improvement of plants. And when you compare all of these methods together, the addition of a single transgene or a single suite of transgenes is by far the most minimal way to go. When we do this, we can tell you where the gene is expressed. We can use very sensitive metabolic methods to determine how different are the two chemically. It's so easy to do these days. And any product that's going to be rolling off the lines at one of the companies, you can guarantee has been looked at those levels, because the last thing they need to do is have something that causes a problem in the population.
One last question from me, and if there's anybody else, I didn't notice hands yet. We heard earlier that there was some evidence of BT proteins being detected in human blood, and I think you said that was not correct.
The study was Eris and LeBlanc, 2011, where they used a kit that was designed for plant biology in human serum. They construct a standard curve, which you can do from their data, which shows the levels of detection of that kit, what the minimum levels of detection of that kit. What they found was 10 to 100 times below the level of detection. So they were reading in the noise of the assay an unreliable amount, which was undetected in most of their samples. In some, they detected, claimed to have detected 12 nanograms per milliliter, which is below what they defined as the detection range of the kit.
And as an order of magnitude, what is 12 nanograms per milliliter? Is it comparable? You give me a...
Well, nanograms per milliliter is the same as saying, you're putting me on the spot here, is the same as saying micrograms per liter, which is the same. So you're talking like a milligram is the weight of an insect wing. You're talking a million times lower than the weight of an insect wing in a milliliter. So just tiny, tiny amounts of stuff. And the detection kits are awesome, and that's what's so good about being able to do this kind of research. If it's there, you can find it. But in this case, they were reading with an unreliable standard and actually reading in the noise of the assay.
And one other thing, I wasn't clear if I understood which way you were saying this. Did you say strawberries are GMO crops or strawberries are not GMO
No, strawberries are not GMO crops. Many people are surprised by that. The only thing are the large ag crops, because the process is so rigorous. We have a strawberry plant that will grow without fungicide. Strawberries are an intensive fungicide crop when we grow them. We have one that uses a strawberry gene in strawberry to make it fungus resistant. This would be fantastic for organic culture. But we can't even begin the process of deregulating that because of the high cost, because of the high thresholds for testing, because we just we can't do it. This would be a wonderful product for us to be able to introduce, but we're completely stymied.
Well, if you'd like to learn how to make the most delicious strawberries, you should visit Boots Hetherington here in Pennsylvania.
I'll have to follow up on that one. I don't know this one.
Thank you very much,
Thank
Dr.
you very
Folta.
much, everybody.
Our final guest today also has traveled to be with us from Washington, D.C. He is Dr. Val Giddings, who's a senior fellow at Information Technology and Innovation Foundation, and you will be speaking from the table. Dr. Giddings, you are the final testifier today. I'm sorry we're running a bit behind, but I think our eviction notice will still be okay. So good luck.
Thank you, Mr. Chairman. It's a privilege to be here with you today. I do note that the time allocated to me from your schedule is minus 5 minutes at this point.
That's right. You were finished 5 minutes ago. You're correct.
So I will brutally truncate the 12 pages of prepared remarks.
We scheduled this with the expectation we might drag a bit from time to time. So please don't feel that you need to compress whatever your remarks are. We need to clear the room by noon, but you're okay. So.
Thank you. I have been working in this area for the better part of 3 decades or more. My training is as a geneticist. I first undertook to work on this area, crops and foods improved through biotechnology, in my capacity as a freshly minted postdoc, PhD, who came to Washington to work for the Congressional Office of Technology Assessment. And 2 weeks after my arrival in Washington, I was briefing then Representative Al Gore on the uses of human gene therapy in medical therapeutics. I stayed with OTA to direct a number of studies, some of the very first studies looking at crops and foods improved through biotechnology, how to do risk assessments, what kinds of policies would allow the opportunities that this new technology afforded to be harnessed with the maximum necessary levels of safety. And so after advising Congress for a number of years, and then spending a year with the World Bank looking at potential applications of biotechnology for developing countries, I then spent a decade with the U.S. Department of Agriculture actually performing risk assessments of some of the very first transgenic crops and foods. And I've worked closely with USDA, EPA, and FDA over the years since then. I also spent a stint directing the biotechnology program, agricultural biotech program for the biotechnology industry organization. So I've worked in government, I've worked in industry. In the last 8 years, I've been working as an independent consultant. I'm here today in my capacity as a senior fellow with a think tank devoted to fostering policies that favor innovation. My interest in this topic, however, is not just professional, it's also intensely personal. I grew up in farm country, although the farm country in Arizona, where I grew up, looks a whole lot different from what you have around here. And although I love the desert, I have to say the beauty of your landscapes is breathtaking. And it's always a pleasure to visit, and thank you again for the opportunity. My front yard was 80 acres of cotton. My backyard was 40 acres of alfalfa and 40 acres of tumbleweeds. And this was all embedded in thousands of acres of citrus, embedded in millions of acres of desert and wilderness. So I'm here first and foremost as a lover of wilderness. I grew up where I'd walk out the back door with my dog and I'd be gone exploring in the desert all day long, and my mother knew not to worry about me unless it was substantially after dark and she hadn't seen me. It was idyllic. It was wonderful. And I retained this passion for wilderness. And in my university studies, it became very clear to me that the single largest impact that human activity has on wilderness, the single greatest threat to wilderness that I love, comes from agriculture. There is no other human activity that has a larger impact on wilderness than agriculture. And so my concern from before I started college, much less a profession, has been in finding ways to improve the sustainability of agriculture, reducing the impacts of agriculture on native lands. But when I became a parent a couple of decades ago, your perspective as a parent changes. And when my son was born 16 years ago, coming up in 2 weeks, we learned when he was about 18 months old that he, to our great concern, has a potentially life-threatening allergy to peanuts. And so my understanding is that your focus here in this educational exploration today has been triggered by the introduction of HB 1770, which would require in Pennsylvania legislation to mandate labels on crops, on foods derived from crops improved through biotechnology. And I'm here responding to that issue in particular because I have a personal vested interest in food labels, the information that they contain, and their importance. There is nothing that will make the marrow of a parent's blood run cold quicker than learning about a mortal threat to a child, particularly one over which you have limited control. We raised our son through grade school knowing that every day he went out the door could be the last time we'd see him because he could die as a consequence of something as simple as a cookie shared on the playground. One of the risks that we could control is that posed by foods that might contain peanuts. Fortunately, food labels carry information to help us make sure that we avoid those sorts of hazards. Much of the conversation about legislation such as HB 1770 has focused on the need for consumers to have access to information about what's in their food. And that's something that no reasonable person would want to argue against. However, the legislation that is before you would in fact do nothing to add to the information that consumers have beyond what they have today, and it would in fact undermine the integrity of food labels in a very important way, because it would suggest the presence of risks where we know there are none. The existing food labeling policy in the United States that is mandated under regulations promulgated or policies promulgated by the Food and Drug Administration require that information that is contained on food labels, this information must be accurate, it must be informative, and it cannot be misleading. They also limit the mandate of information contained on food labels to information that is relevant to health, safety, or nutrition. So if something is relevant to health, safety, or nutrition, it must be on the label in a way that is accurate, informative, and not misleading. As you have heard from earlier speakers, including Professor Ott's excellent disquisition, the information that something has been derived through biotechnology really tells you absolutely nothing about the safety of that product. Indeed, in fact, derived through biotechnology, or GMO, is a process-based label that tells you nothing about the contents of the food on the label. If, in fact, the proponents of this legislation were serious in their desire to convey useful information, they would require not just information about the process, but they would require that the label contain specific details about the particular construct, the particular novel protein that would be in the food involved, but that's not what they're calling for. The reason for that is very simple. This demand for mandatory food labels for GMO-derived foods is not about informing the consumer. It's about promulgating a fear-based marketing campaign that is in fact designed to expand the market share for non-biotech-derived foods, in particular organic. Consumers today have the option to choose foods that have been derived through methods other than biotechnology. They can buy foods labeled organic. They can buy foods certified through the non-GMO project. They can download apps onto their cell phone so they can scan the barcode of a food in the grocery store to tell whether or not it contains ingredients derived from crops improved through biotechnology. The information is out there to the consumer. Consumers have plentiful access to it. The motivation of those pushing this legislation has been very clear. As I said, it's part of a fear-based campaign through which they seek to expand their market share. Kevin Folta included the pictures and names of some of these individuals. Let me give you some quotes about their motivation in these campaigns. We're going to force them to label this food. If we have it labeled, then we can organize people not to buy it. That's from Andrew Kimbrell, who is the principal author of the kinds of legislation that you are considering on this topic. Joe Mercola from Mercola.com, who makes boatloads of money peddling alternative medicine cures, has said, personally, I believe GM foods must be banned entirely, but labeling is the most efficient way to achieve this. Since 85% of the public will refuse to buy foods they know to be genetically modified, this will effectively eliminate them from the market, just the way it was done in Europe. Jeffrey Smith, who has written and self-published a number of volumes containing some extremely imaginative fictions about the potential dangers of crops and foods improved through biotechnology, has said, by avoiding GMOs, you contribute to the tipping point of consumer rejection, forcing them out of our food supply.
What he's saying. This is an opinion. Sorry, Karen. I object. I object. I'm not a fear mongerer. I'm a scientist. Yeah.
With labeling, Vandana Shiva has said,
to all in this room, there have been personal points of view expressed by many of the folks testifying to that. I have asked everyone to state the science that has not proceeded in that fashion throughout. Since I'd say our second testifier, the first two were purely science. But I understand people have points of view and there's some debate, and that's why you're objecting. But this is a hearing, an informational meeting, where we invited everyone from those who have been advocating for this bill identified as being a scientist to speak. Every single one was invited. And those that could attend did, and we're grateful for that. We've also asked for other suggestions as to who testified. And I think we should show the same respect to those who come who may not share the same point of view as they showed when the others were speaking. So I would ask for no further interruptions, and Dr. Please proceed.
Continuing to quote those who are driving the proposals for mandatory labels, I would point out that Vandana Shiva has said with labeling GMOs, the percentage will become 0. For you, the label issue is vital. If you get labeling, then GMOs are dead end. Ronnie Cummins from the Organic Consumers Association has said, the burning question for us all then becomes how and how quickly can we move healthy organic products from a 4.2% market niche to the dominant force in American food and farming. The first step is to change our labeling laws. I'm delighted to join Chuck Benbrook in noting that the foods derived from crops improved through biotechnology on the market today are safe. I think that the kinds of correlational analysis that you have seen, some presentations of attempting to blame glyphosate for a series of ills, is in fact irrelevant to the issue of labeling for foods derived from crops improved through biotechnology. I'd be delighted to talk about those issues. I'd be delighted to talk about colony collapse disorder and beekeeping and crops improved through biotechnology. Any range of questions that you may have, I'd be more than happy to respond to. And I thank you again very much for giving me the time to talk this morning.
Thank you, Doctor. Going back to sort of helping with the scientific understanding, I'm not involved with the FDA, or obviously it's state, that's federal. There was discussion earlier that the FDA has not concluded any GMO foods are safe. It's gone through this consultation process, which apparently is voluntary. But the question I have is, when you were talking about organic labeling earlier, does the FDA conclude that organic foods are safe?
No, sir. The FDA has nothing to do with the organic label. That's a marketing program and a marketing tool administered by the Department of Agriculture.
Just demonstrating, I don't have, we're learning.
Sure.
Now, the other question is, does the FDA approve any food as safe? For instance, I become more mindful of what eggs I buy. Because it depends what you feed the chickens, at least to what the eggs are. So in a sense, I suppose that's genetically modified. But if I'm a chicken grower or egg raiser, do I go to the FDA and ask them to approve my eggs as safe?
Well, again, eggs generally are reviewed for safety by the USDA. But let me address your question about FDA and food safety and what they do and what they don't do. Dr. Benbrook was entirely correct in saying that FDA has not concluded that any biotech-derived food is safe. But it's easy to misunderstand what that means. The reason FDA has framed the language that they use in the so-called voluntary safety review process, consultation process that they make available to the developers of these foods is because they want to make it absolutely clear to those developers that those developers bear the legal and moral responsibility to make sure that the food that they put on the market is safe. What FDA does is FDA administers a law, and that law requires that all food placed on the market be safe. Now, FDA doesn't, as USDA does, put an FDA good housekeeping stamp of approval, this is safe. What they have said is that They follow the National Academy of Sciences and the OECD, which has set international standards for the type of risk assessments that you use to evaluate the safety of these products. FDA follows the policies that were laid down after a decade of consideration and exploration on these topics, and they have made available a series of questions that developers of these foods need to provide answers to make sure they meet their obligation to produce a food that is safe. This process that they go through, this consultation process, is a voluntary process, but it is in fact de facto mandatory because no company would be allowed by their corporate counsel to put a food on the market that has not gone through this consultation process. So it is de jure voluntary, it is de facto mandatory, and every company that is, every individual, every food that's been placed on the market has gone through this consultation process. There are no plans for putting any foods on the market that haven't gone through it.
Were there foods that have been presented to FDA for this process that have subsequently been determined to be unacceptable?
No. No, there was one case where Pioneer Hybrid was developing a soybean. And they wanted to develop a soybean to produce a better poultry feed. Because poultry feed, soybean is a very good protein source, but it's deficient in the sulfur-containing amino acids like methionine. And so poultry farmers typically had to add some supplements to the soybean-based poultry feed to make sure that the poultry they're raising got enough of that key amino acid. Pioneer identified a protein from Brazil nut that produced sufficient amounts. They put it into the soybean, and they found to their dismay, long before they even began the consultation process with the FDA to put it on market as animal feed, they found to their dismay that in putting this gene, or Brazil nut, into peanuts, they had also transferred the gene that the allergenic potential of Brazil nuts and made those soybeans allergenic. So they elected not to bring that to market because of all the potential obvious complications. I have colleagues in Brazil who said, give it to us. We will gladly put this on the market and accept the potential risk for human allergenicity in exchange for a better tool to conflict malnutrition in the poverty-stricken areas of northeastern Brazil. But Pioneer did not give it to them, and folks at Embrapa in Brazil are continuing to try to develop that improved type of soybean today. But there have been no food consultations in which FDA has responded by saying the information you've provided has not been sufficient. We are not confident in the safety of this. Don't put it on the market. There have been none such. And there have been none by FDA comparable organizations elsewhere in the world.
Representative Morgan Beto.
Thank you. Thank you for your testimony. Honestly, what I'm trying to understand is, would you oppose all labeling? You say that HB 1770 is not a good, but you would certainly want to know if there was peanuts in something for your child, right?
Yes, and existing law requires that.
Right. And so I guess what I'm asking is, what would you suggest as the proper labeling so that people have the information in a way that doesn't just feed the concerns about hysteria, but allows people to make a choice, just as you're making a choice and being proactive with your son, until science catches up and is able to educate everyone? In other words, what would you suggest as the legislation?
Well, I think the gold standard for a food label that's required by government, I think the gold standard would look very much like the label that FDA requires today. A label is required to carry information that's relevant to health, safety, and nutrition, and it's required to present information in a way that is useful, accurate, and not misleading. I think if you can meet those criteria, then that's going to be a pretty good food label. And there's a lot of different ways that you can present that information. We've seen FDA and USDA working together to redesign food labels several times in the last decade or two. And I think that these have produced labels that have incrementally gotten better. But there are some types of things that some folks want on labels that can't meet those criteria. Some folks want to know whether or not something has been produced with recombinant DNA techniques. You know, the term GMO, I think, as a geneticist, I'm really unhappy with that term because, as you have heard, as Dr. Folta pointed out, as others have said, everything that appears on a dinner plate any place on the planet is genetically modified. Even wild fish have been subjected to artificial selection by humans for generations, such that the average size of trophy fish from the Gulf of Mexico today is about a quarter of the size that it was 40, 50 years ago. You know, we have altered genetically the makeup of virtually every species of plant and animal that we eat, if not every species on the planet. Everything living on the planet has been genetically modified one way or the other. And the techniques that we scientists use in the lab to produce what we call GMOs are in fact techniques that we have discovered by finding them operating in nature to affect genetic change in nature. So these techniques that we use are directly comparable to those that we find manifest in nature. The single most common gene in a human genome, in each and every one of us in this room today, is a gene of viral origin. It came from a virus. It can't, in any sense, be called a human gene, except for the fact that it's the most common gene that we carry.
Well,
You know, so people who want to have information, you know, like, is this GMO according to this artificial distinction that's not supported by science? How can you meet that kind of a need? I think that it's already met through the organic label, because people know that organic production methods prohibit the knowing use of foods, of crops improved through modern recombinant DNA techniques or biotechnology. You know, there are market-based and voluntary labels that can be used in a way that is not misleading and that is not scientifically accurate, ways that do not suggest the existence of risks where science has shown they don't exist. The only safety differential that the scientific literature has ever demonstrated between a food derived through biotechnology and other sorts of food has been to the benefit, showing that the biotech-derived food is safer. And that's come in the case in particular with corn, BT insect-protected corn, because they have 10 to 100-fold lower levels of contamination with cancer-causing compounds caused by fungal diseases, which the biotech trait eliminates. You know, so the safety of, as Dr. Folta said, this is not a scientific discussion. This is something else. You know, so what are the other ways in which you can meet those needs? I think there are a variety of voluntary efforts that can satisfy the need to know of folks who have these concerns, whether or not they are justified on a factual basis. But I think to mandate a label that would mislead consumers about the safety of these products, as this well-funded campaign that is pushing for these labels across the country does, I think that would be bad policy.
All right. Well, the good news is we have 2 minutes before our eviction is required. There's another meeting beginning in this room shortly. I had promised we would have cleared out by noon, so I will ask that once we are adjourned that we orderly proceed outside so the folks setting up for the next meeting can get about it. So thank you all who traveled today. Thank you for the wisdom. I think everybody who attended learned something from every presentation, and I very much appreciate it. We're adjourned.